fine. Let the robot do it. Okay, hello everyone. Thanks for everyone who survived and joined us on the third and last day. I'm very pleased and excited to invite Jonathan Nachensson for our first keynote talk for today. Jonathan completed his PhD at the Hebrew University in Jerusalem. I was gladly his first postdoc opportunity, which was great. Jonathan actually helped me set up my lab, and he's responsible for the fact that we actually have our first experimental paper. And thanks for not giving up, so that's good. Jonathan then continued to the Technion, where he actually worked with rats, or mice. Was it rats? Mice. Well, same. They both have tails. So working a lot on memory. So Jonathan does both philosophy of neuroscience and actual neuroscience. So I think these are the sort of people that we want to see more out there. And yeah, now he's a Minerva Fellow in Bochum, so a local. And he's going to talk to us again about memory today. So specifically mapping memories, can we reconcile the spatial and mnemic conceptions of the hippocampus? Please, Jonathan. Thanks.
Thanks for the warm introduction, and thanks the organizers for putting up this wonderful event. So what I want to do today, I want to talk with you about two ways of understanding what the hippocampus is doing. One is what I call the spatial conception. So the hippocampus is in the business of computing cognitive map. And the other conception, which I call the mnemic conception, is that the hippocampus is required for episodic memory. And basically my talk will focus on how and if we can reconcile these two different conceptions of the hippocampus, which along the history of research on the hippocampus was conceived as being two opposed conceptions. Okay, so it has long been recognized that the mechanisms that produce episodic memory and those that transform and encode the spatial information share many similarities, perhaps being the very same mechanism. But much of this debate has been built on two non-converging at times bodies of evidence. So a lot of the evidence for the role of the hippocampus in memory comes from neurophysiological lesion data, most famously HM, but also other patients, which suggest that all of these patients suffer from different forms of amnesia, but still can perform basic spatial tasks. Another body of evidence comes mostly from animal studies and finds evidence primarily for spatial representation, and culminated in a Nobel Prize given to O'Keefe and the Mosers for finding place cells and grid cells, but there are other spatially selective cells that are involved in the hippocampus. So we can very roughly divide the views of the function of hippocampus to three major camps. This is somewhat of an idealization of the theoretical landscape, but I will use it as a baseline for my talk. So one camp take it that the hippocampus is computing a spatial representation in support of cognitive maps. We can define cognitive maps as an allocentric representation of spatial environment and the layout. This is thought to allow flexible navigation and planning, most famously suggested by O'Keefe and Nadal in their book from the 70s, but later picked up and developed much more by David Reddish and the Mosers with their idea of a metric in grid cells. Another camp takes the hippocampus to be in the memory businesses. So this is, so the function here is considered to be something like the generation of episodic representation of the personal past, or something a bit more generally, also including counterfactual cognition in general. And this is something offered, for example, by Donna Addis and Kirk McCallion, which takes the hippocampus to be an episodic simulation system. A third camp, which I'm not really going to talk about today, but we can pick it up in the Q&A, consider an alternative function, like something like relational memory. The most famous proponent of this approach is Howard Eichenbaum, which so that you can use this idea of relational memory as something that can be responsible for both spatial and episodic representations. So so I'm going to be focusing really on those kind of two camps, the cognitive map camp and the memory camp. And traditionally, these have been seen as two opposing views on what the hippocampus has been doing. But this opposition between cognitive maps and memory has been reevaluated recently, where current views emphasize that cognitive maps are not just parallel to memory or even have to do with a different kind of functional role that the hippocampus has, but are really integral to how we understand mnemic functions. So these views highlight how the structure and how spatial structure and organization can be used to explain phenomena that are related to episodic memory, like semanticization, consolidation, and graded amnesia. So according to Sarah Aronovich and Lynn Nadal, who have been pushing for this kind of view, this is due to how spatial coding schemes are used to organize and generalize information. So one idea that they kind of offer is that we can return to the same place multiple times, which makes spatial information repeatable. This is like location queuing heuristic, as they call it. And that allows organisms to generalize information coming from multiple experiences because it is bind into the same spatial location. Another type of suggestion along those lines has been put forward by Ellie Boyle, where she basically considered the function of episodic memory in spatial terms. So she offers that we can think about the function of episodic memory as providing a spatial scaffold for semantic learning. And she takes her inspiration from the method of Loki. So perhaps some of you will be familiar with this idea of memory palaces, which are a tool in mnemonics, okay? So this idea of a memory palace is well suited for the aim of organizing information because it can provide you a stable landmark that is useful for organizing experience. I can remember a central fact if I have a mental model of where is that kind of information located near to, or in what place in my spatial organization scheme is that information is located. So according to these kind of philosophical views that try to integrate these two conceptions of the hippocampus, spatial representation serve to organize encoding, storage, and retrieval of learned information. So they take cognitive maps to be for memory. We can understand the functional relation between cognitive maps and episodic memory in two ways. One is to kind of emphasize the gradual nature and to kind of elaborate on the evolution of the hippocampus. So this idea coming from Buzaki and Moser is that co-opting the evolutionary development of egocentric path integration. I think that Peter had mentioned some of this work in his talk. So these are kind of phenomena that already appear, at least dead reckoning, appear in insects. So co-opting this kind of mechanism or the evolutionary development, more specifically from egocentric path integration to allocentric map-based navigation, mechanisms of memory evolved from mechanisms of physical navigation or of navigation in the physical world. But this isn't just an evolutionary story. So spatial organization is thought to provide a computational framework. And the idea here is, and this is like work by Whittington and Behrens and many other computational neuroscience these days, is that the same underlying mapping structure can support both navigational and memory representations because spatially structured representations share common organizational principles. So the same way that we can think about the relation between, the spatial relation between landmarks like north, south, west, etc., we can also think about the spatial organization that exists, for example, in a family. So the relation between a father and their children, grandfather. So there is also this kind of structural generalization that goes on in many ways in which we can conceptualize a relata. So this idea gives the impression that not only are these conceptions are opposite to one another, they are really integrating the spatial and mnemic conception of the hippocampus is really desirable and even achievable in practice. So what I kind of reviewed so far is this idea that cognitive maps have been proposed as computational vehicles which, in virtue of their representational role, play a part in encoding, organizing, and retrieving learned information. But, and this is basically what I'm going to focus moving on, is that determining whether cognitive maps are relevant to the function of memory reveals a tension in how we delineate the cognitive architecture of the hippocampal system. So what I'm going to argue is that mechanistic decomposition of cognitive maps and any attempts to basically import the mechanisms underlying cognitive maps suggests that some of the functionally relevant components of cognitive maps are either not components of memory systems. So if we really want to integrate those mechanisms, that will require a revisions of the explanans, the mechanism providing the explanation, or are irrelevant for episodic memory. So if we really want to understand and explain episodic memory by cognitive map, that will require some revisions of the explanandum, the phenomena that we are aiming to explain. So the plan of the talk is as follows. I'm gonna present what is mechanistic decomposition as a mechanistic strategy to understand systems as mechanisms, and I will kind of talk about how has this strategy been applied to cognitive maps. This will lead me to kind of consider, okay, how do we understand the relevance of the hippocampus as a component in cognitive maps vis-a-vis episodic memory. This leads to a dilemma, I want to argue, and the third part of the talk is basically trying to flesh out the horns of the dilemma and some of the trade-offs that are involved in picking one of the horns. And the last part of the talk is trying to offer two ways to avoid the dilemma and perhaps showing some of the trade-offs that are involved in picking those escape routes. So what is mechanistic explanation? So some of this material is already, I think Alexander already kind of reviewed some of this stuff, but to have like a common baseline between all of us today, I'm gonna repeat some of it rather quick. So mechanistic explanation describe the underlying mechanisms that give rise to an explanandum phenomena. This type of explanation treats the explanans as a mechanism, components organized in a way that produces the phenomena. And mechanistic decomposition is basically a strategy that we apply to systems when we want to understand them as mechanism. It is the process of breaking down a system into components to understand how the mechanism produces the phenomena. So if we want to understand how does the car has the capacity to drive, we break the car into its constitutive components, its engine, its radiator, its battery, and by explaining how are these components organized with one another, we can explain how the car is able to take us from one location to another. So an important aspect and something that isn't so much discussed, at least in the classical literature on mechanism, is that the similarity between mechanisms depends on their shared components organization. So, for example, while motorcycles and cars have core components, they have engines and batteries, motorcycles lack reverse gears. So while they both have a capacity to take us from one location to another to perform driving, they do it by somewhat different mechanisms. So this same explanatory strategy, the mechanistic decomposition, has been applied to understand how the brain generates cognitive maps. And this decomposition revealed that an agent's ability to perform navigation tasks, at least of the sort that the hippocampus makes possible, depends on the interaction between different spatial representations. So there are different ways to decompose cognitive maps. Here I'm going to primarily rely on on David Reddish decomposition of cognitive maps, which I take to be the most elaborate and detailed account of that. But there are also less detailed accounts. Sometimes cognitive maps are used in a more metaphorical way, even without any relation to their underlying mechanism. It's also a construct in psychology where the mechanistic underpinning may perhaps be less relevant. So I'm really talking about the underlying mechanism of cognitive maps and this way of decomposing cognitive maps. But in general, there is some consensus that most of these decompositions will be quite similar and will have at the least these kind of minimal components. So under this idea of decomposing cognitive maps, they are thought to be composed of the following components. So you have to have a mechanism that is responsible for a local view. So that will be a mechanism that computes a representation of the animal relationship to landmark in its environment. You need to have some form of a distance metric, so a component that computes a representation that encodes the distance between locations. So this famously has been thought to be something that grid cells in the medial entorhinal cortex are doing. And you need to have a head direction mechanism, so a representation of orientation in space, and these were the head direction cells famously discovered by Rensk. So you can see, like, we represent the direction of to which the head direction are sensitive by using like this some kind of a polar coordinates. So this is a mechanism that is responsible. Okay, where in space am I oriented toward? Another component is the place code. So this is a representation of self-location that ties those local views to a distance metric. Where am I now in comparison to the landmarks that I'm perceiving and my distance from them? And a goal code. So that will be a representation that associates motivational and spatial inputs to plan a trajectory. So where do I want to go, given the location I'm at right now and the landmarks around me? Okay. So although there is this kind of, perhaps a caricature that the hippocampus is intimately tied or even like completely responsible for cognitive maps, this has never been the claim in cognitive map theory. So the hippocampus is not considered to produce cognitive maps in toto. According to cognitive map theory, even as originally proposed by O'Keefe and Nadal. Its functional role is to compute a self-localization signal within an allocentric spatial representation. So according to cognitive map theory, the hippocampus computes a representation of position, a self-position, that ties perceived landmark to distance and direction. So in that kind of supermarket list that I gave you about all the components involved in the decomposition of cognitive maps, that is like the place code, the hippocampus, at least CA1 or CA3, that's like thought to be involved in computing a place code, and this is like those famous place cells that fire when the animal is in a specific location. Okay, so after kind of going over this idea of the componential role of hippocampus, I want to understand, okay, if that's what we are taking the hippocampus to be doing, how do we understand its relation vis-a-vis this attempt to integrate cognitive maps and episodic memory? So, as I said, according to cognitive map theory, maps are constituted by multiple components. So distance encoding, self-localization, goal representation, et cetera, et cetera. However, taking this theory to explain episodic memory raises key question about how we characterize the hippocampus as an episodic memory system. On the one hand, if we take all components of cognitive maps as required to memory, then the relationship between the hippocampus and memory becomes a bit more tenuous. A lot of other anatomical and functional structure and circuits are required to compute cognitive maps. So you will require parietal mechanism to basically encode your relation to landmarks. You need like reward circuit, nucleus accumbens, the ventral tegmental area in the midbrain and the basal ganglia to compute some form of goal representation, so something to integrate the motivational spatial inputs in order to plan this kind of, okay, where do I want to go? So a lot needs to change if we really want to take all the mechanism of cognitive maps at face value. On the other hand, if only a single component of maps, and this is basically, I think, like somewhat of the traditional view, the idea of a place code is needed for memory. So cognitive maps are not strictly speaking for memory, at least in this mechanistic interpretation. They do not play the same functional role as they do in navigation, where functional decomposition revealed that we actually need all of those components in order to compute maps that is needed for solving those allocentric base navigation tasks. So this leads to the following dilemma. So specifically, how we carve the relation between cognitive maps and episodic memory leads to a dilemma. Either we extend our understanding of, or the construct of memory beyond the boundaries of the hippocampus, involving other neural systems, even like non-conventional declarative memory circuits, or we go specific. So after seeing Oron's talk, now I kind of think to myself that it follows the same kind of structure between the general and specific that Oron was mentioning yesterday. So either we go specific, and then what is important is not cognitive maps per se, but some specific computation, computing some self-localization signal, which can be used for cartographic and mnemic aims, given the demands of the task. Either horn we eventually pick, however, comes with its own epistemic benefits, but also with its own epistemic costs. And in this part of the talk, I want to kind of flesh out the horns of the dilemma and the costs associated with picking one of them. So according to the Broadhorn, if we really want to kind of integrate this kind of spatial computational structure to understand how does the hippocampus solve episodic memory-related tasks, then we need to include a lot of the components that are needed to explain those rich spatial structures that are made possible by cognitive maps. But if all components of cognitive maps are relevant for memory, then we might need to revise our conception of what is the function of episodic memory. So, for example, if memory involves the goal-directed computation seen in spatial navigation, selecting paths to reach specific goal, or defining a goal location in abstract space, then memory too becomes enmeshed within systems that fulfill functional roles that go beyond remembering. So adopting this broad horn may be seen as too revisionary to some, as it involves a lot of reconceptualization of what we understand as essential to episodic memory. So this can be cashed out in two ways. One is that, and here, for example, I'm gonna focus on trying to incorporate reward circuits, for example. So one is this, is that memory becomes much more goal-driven. Goals are intrinsic to cognitive maps. One can say that are intrinsic to any navigation task whatsoever, but this is debatable and controversial, so I'm not going to. But in order to plan a trajectory in cognitive maps, you need to have a goal. When we plan how to get from point X to point Y, we inherently identify Y as the goal. So this relates to two types of representational content. So we can think about the representational content as descriptive. It represents how things are, can evaluate by some measure of accuracy, can be more or less correct. Another way to think about representational content is as directive. It instructs the system on what actions to take, and it has the function of that causes the consumer to behave in certain ways. Memory representations, if we're really going to be serious about incorporating all this mechanistic architecture of cognitive system, then it will need to reflect the directive nature, because in cognitive maps, this is the functional role that like a goal representation has. It is a directive representation. It instructs the system on how to plan a trajectory to get from point X to point Y. Memory then becomes, it kind of distance from perhaps our folk, but even I think a traditional psychological conception of what memory is, about something like about tracking the path, and more even about something like motor planning. The second way in which we can cash out this idea is through recharacterization of memory system. What is recharacterization? Recharacterization of a mechanism refers to the process of reconceptualizing, refining, revising the components and the organization of a mechanism to better explain the phenomena. Thing is that both classical and even more recent framework of memory systems, so what we see here is like the more traditional one, in which we divide long-term memory to declarative and non-declarative memory, and we have episodic memory and semantic memory in circuits that are in the temporal lobe, while we have all the other stuff. Usually, this is like the criticism. All the other stuff we put in a wide variety of brain regions, like conditioning, priming, sensitization, and procedural memory. So, but even like, even more recent that tries to, I don't know, identify procedural memory as having like a much more cognitive character, they still typically separate the episodic memory system from reward circuits. But having a goal isn't just about searching an event in memory, it's really about how reward circuits actively shape how information is organized and accessed. So this could mean that reward circuits might not be just modulators of the inputs that are being processed in the hippocampus, but becomes really constitutive components of the memory mechanism itself, which will lead to a recharacterization of the episodic memory system and even the entire framework of memory system. Okay, So if we have all these kind of problems or issues, if you like, with going broad, how about going narrow? And so according to the narrow horn, what's important is not really everything that cognitive maps are doing or the mechanism underlying cognitive maps are doing. It's a specific computation. This is basically the more traditional, classical approach to understanding the functional role of hippocampus in the context of cognitive maps. And that, depending on the test that the animal needs to solve, we can understand it as playing a cartographic or an anemic role, depending on those test demands. One, I think, big benefit of adopting the narrow horn is that it has the benefit of turning the incompatibility between cognitive maps and episodic memory to merely an apparent one, something that basically we theoretically contrive. And this is, I think, there's the theoretical advantage of dissolving decades-long debate on what the hippocampus is doing. But it also has some issues. One of them is that much of the theoretical richness that I alluded to in the beginning, those kind of structural organization scheme, those are potentially lost. So one implication is that those theories on which the function of memory depends on spatial structures are really very limited, because the episodic memory system might be functionally distinct from the spatial coding schemes that are used to organize storage and retrieval of learned information. Another thing is that the mismatch between explananda still remains. We still need to explain phenomena associated with either spatial navigation or episodic memory, and how does the hippocampus switches between those depending on task demand. Saying task demand by itself, it does not provide an explanation of the involvement of the mechanism in two different contexts. So how should we understand the kind of trade-offs that are involved in this horn? So an important point is that there is some form of functional asymmetry between spatial and episodic representation. So while spatial representations are only part of the representational content of episodic memory, spatial representation can exist independently of episodic representation. As I said in the beginning, this is actually one of the major drivers of this opposition between episodic memory and cognitive maps conceptions. So a lot of amnesic patients suffer from various forms of amnesia, but some form of, they can still perform some forms of spatial tests. So it seems that there is a way in which we can understand how the brain is computing spatial representation without saying anything about episodic representation, but it's not very clear if we can go the other way. So the dependency here runs one way, so to speak. But this idea of spatiality probably exists on a spectrum, so episodic representation can be more or less spatially rich. I can remember thinking about these kind of issues early this morning, and this doesn't involve any substantial reference to space, at least not to the landmarks or to the location I was inhibiting. But I also can recall traveling around Germany in a beautiful landscape, and those are the kind of memories that involve a lot, that have a rich spatial content. So, like, even in our memory, we have something that is much more minimal, and those kind of memory that are really spatially rich. But even if we acknowledge that there is this kind of spectrum, it still seems that we remain caught in the dilemma for those rich spatially representations. So these are we in a case of cognitive map for the spatially rich? This could be some way of avoiding the dilemma, but or kind of barricading it for a special case of representation about the past. Even if we want to maintain episodic memory as a unified construct, we still need a story that accounts for how those spatially poor and those spatially rich episodic representation fit together, and how can we move in what seems to be a unified mental experience between the spatially and the spatially rich and the spatially poor. So I think that the narrow horn doesn't, it's not only about losing the explanatory richness that comes from cognitive maps. It reflects constraints on how episodic memory needs to be explained. It has to have some kind of reference to how spatial information, spatial input, and spatial signals play a role in how we are able to remember the past. So while this horn has the benefit of dissolving this apparent conflict, it sacrifices a lot of computation that might seem necessary to explain the function of memory. So in the last part of my talk, I want to offer two ways in which we can avoid having to pick perhaps one of the horns or escape the dilemma. And try to point at some issues that even trying to escape has its own cost, so to speak. So one way is to emphasize the continuous nature of the function of hippocampus. So this is trying to provide an evolutionary story by saying that the hippocampal spatial computations in non-human animals are co-opted or reused or expanded for episodic representations in humans and even in non-human animals. We saw something along that approach at the start of the talk when we considered the evolution of path integration mechanism to allocentric map-based navigation. So here we don't face this issue that we need to reconcile two different conceptions, is that, okay, this idea of cognitive maps explain a lot of animal behavior, but perhaps when we move to what's often called higher level organisms, we start to attribute much more complex cognitive structure. So what we are changing basically are the species, but the kind of computations remain the same in a sense. So this raises what I call, appealing to this kind of co-option argument raises what I want to call this inferential domain problem. I didn't see much discussion of it in discussion of exploitation. So, but if anyone has an idea if it's already been discussed and under a different heading, please tell me. So this is the idea that we keep using evidence from instances of the original function to make claims about the co-opted novel function. So we use evidence from spatial navigation to make claims about episodic memory. But if we're really talking about a co-opted function, we really need independent evidence for each domain. We really need independent evidence for how does the function behave in the new, following the adaptation in a different environment. So. To kind of illustrate this idea, I want to look at hippocampal replay as an example. So hippocampal replay has been used to explain both, computation that are related to both spatial and episodic representation, and also a lot of the time the way that philosophers and also neuroscientists talk about replay still. Although there is this kind of assumption that replay behaves or was co-opted for other purposes in humans, they still appeal to evidence about the way that replay behaves in rodents, for example. So I want to kind of show the issues that arise from us trying to maintain all these bodies of evidence together. So what is hippocampal replay? Hippocampal replay is a sequential reactivation of neural activity following experience. So the rats travel along a path in the maze. He stops, or whether he goes to sleep or stops for making a decision. And basically, we can decode a sequence of the places that the animal transverse. So this can happen, as I said, online, or it can also happen offline. A lot of the evidence that goes on in humans comes from offline studies in sleep. But, and it manifests as at least in electrophysiology of rodents, but also in calcium imaging, as a short burst in the hippocampal signal, also known as shortwave ripples. So, as I said, while replay exists in both humans and in non-human animals, there's a lot of significant differences that make it problematic for us to keep using the evidence that we find in rodents to extrapolate to humans. So, in non-human animals, sequential reactivation of places represents past and potential future paths. But in humans, it's actually less tied to spatial experience, operate on something that is sometimes called factorized representation. So for example, for animals, they use replay to plan a path for a food location. But in human, replay separates aspects of an experience to a learned rule, such as I can find a good schnitzel in the Black Forest. Okay, so like what are the common transition structure that exists among many spatial trajectories, even to the point that they lose a lot of their spatial information. But the differences are not only functional, they are also mechanistic or physiological. So ripple band power increases observed in humans during replay events happen at much lower frequencies that compared to rodents. This could be because humans have a bigger brain, but those kind of issues about the transmissions of signal play a role in the type of phenomena and mechanisms that are supposed to explain the phenomena. Another, I think, more striking difference is that replay happens after exploration in rodents, but it happens prior to retrieval in human and non-human primates. So it is used as some way to predict the next step in a sequence. So basically, the temporal position in the causal change is different between those species. So those species-specific differences in replay, I think, illustrate the inferential domain problem, as I called it. We can't, okay, so yeah, okay, I'm about to end. We can selectively use positive analogies while rejecting the negative analogies. You can get to pick and choose whatever you want. Basically, one needs to be consistent of the type of evidence that can be applied across species. So while co-option is plausible as a story to explain, there's a lot of homologies. We saw a lot of talks during the conference of those sophisticated mechanisms that exist across species. It leads us in some way back to original dilemma. If we focus on the differences between species, we're back to revising perhaps the function of episodic memory. Perhaps it's much more related to abstract rules, closer to semantic memory than to personal past experiences than we tend to initially believe. If we opt for the similarities between species, we are limited in the evidence that we can draw from spatial cognition in non-human animals. So this kind of dilemma, this is a broader issue about extrapolating from model organism, but it also has this kind of special case when we try to integrate the spatial anemic conception of the hippocampus. Okay, yeah, I think I'm gonna be able to do it. Okay, so if continuity poses problem, why can't we consider the function to at least partially overlap, sharing some components but not others? And I think this has been the preferred approach in cognitive neuroscience, where the hippocampus, specifically the dorsal or the posterior hippocampus in humans, serves as a joint hub in a broader, partially overlapping neural network. So this can help us avoid this idea that we really need to incorporate the entire underlying architecture of cognitive maps. But it faces, I think, two issues, is that when decomposing this context-sensitive system, we must account for context switching. We need to find those parts of the mechanism, those network parts that reinstate context, and those that implement task-specific organization. And those, almost by definition, will not be the hippocampus. So what will be determining if the hippocampus is remembering now or not involved or not engaged in solving a remembering task will not be the hippocampus, will be another component that basically allows you to switch between those kind of regimes. Another issue is that partial overlap indicates that the computational operation predicted by cognitive map theory cannot be directly imported, as the functional profile of a component can change due to the constraints of the network. So just by placing the components in a network, it can change its own, its own functional profile. This leads to somewhat like an unhappy marriage in a sense. So the separation between network extends memory beyond the hippocampal complex, but separating spatial and non-spatial retrieval creates these challenges for integrating this computation. So while we expand memory beyond the hippocampus, we still face those burdens of explaining how these separated processes could support this spatially integrated episodic representation. So we try to escape the broad horn, but we import a lot of the theoretical tensions that we have from the narrow horn. So to conclude, throughout this talk, I explored all these kind of ways to reconcile the spatial and mnemic conception of the hippocampus, putting my emphasis on those more recent offers to that these conceptions are not in opposition, aren't parallel even, but really cognitive maps are integral to how we need to understand the function of memory. I argue that this leads to a dilemma. Both horns have the benefit of resolving some of the tensions that these two views of the hippocampus had, but each comes with its own cost. If we want to include all the architecture of cognitive maps, that this is the architecture that provides us those spatial coding schemes, this may lead us to recharacterize how we think on episodic representations and a memory system. If we focus on specific computations, this sacrifices theoretical richness, the type of richness that those kind of recent approaches appeal to, and it also faces the challenge of explaining both spatial, episodic, and spatial and episodic phenomena. So I want to emphasize that I don't take this kind of dilemma to be too bad alternative. This is really more a case of an embarrassment of riches. These are theoretical choices, each with its own costs and benefits. There is not really a stalemate here, just basically theoretical roots that will lead to some implication down the road. So whatever theoretical route one takes, I think that careful attention to mechanistic decomposition and trying to import something beyond the computational structure, but also the mechanisms that are responsible for those computations, really pushes us to reconsider the function of the hippocampus. Thank you.
Thank you very much, Jonathan. Questions? David, please.
That was a very nice talk, thank you. At the outset, you framed the discussion by saying you weren't going to talk or consider other conceptions of hippocampal activity.
Yeah.
But then you point out the difficulties in trying to meld these two different views.
Yeah.
And I just wonder if, isn't there a reasonable response here, which is just to say, well, maybe we do need to consider sort of unified conceptions. So I'm thinking in particular of the optimal compression hypothesis from Fusi, where he, very complicated mathematical theory about hippocampal function, about optimal compression. And they have all kinds of results that are consistent with episodic memory findings as well as navigational findings. So wouldn't that be a more reasonable sort of outcome than trying to hacksaw your way? Yeah. So what do you think of that?
The optimal compression, like I think some leading candidates, so I have a cheat slide. A leading candidate today, and this is stuff that we did in my previous lab, is to consider state transition model. So, work by Stachenfeld and Gershman. So this is like they encodes the states and the likelihood of transitioning between states. So these kind of approaches also has a lot of merit in trying to go beyond, but I think by themselves, so stuff that I talked about in relation to making memory much more goal-driven, they already, so in order to kind of compute a predictive map, you have a value function, you already insert all this kind of incorporation considerations, but it also, one point that I think is a remaining worry in trying to kind of appeal to those kind of models, I don't know the optimal, we can talk about it later, it sounds interesting actually, is that you really move to from a more physical space to a more abstract conception of space. So this is actually a point made by Sarah Robbins and Sarah Aronovich that it's not clear whether we are still talking, we're still holding to a literal conception of space or something much more metaphorical. So in order to compute cognitive maps, a lot of embodied elements are important. You have to have self-movement cues and optic flows, which don't have any way in which they can appear in those state transitions. You already include other mechanisms that will take you, again, to adopt this kind of, we need more of the architecture of cognitive maps. So in order to, how do I say, in order to instantiate the models in a way that can allow you to explain physical navigation, you already will try to incorporate more. So it puts you in this kind of abstract position, which I take to be good to unifying, but you also lose a lot of the type of phenomena that you can explain. Yeah. But I don't know about the optimal stuff, so perhaps everything I said was wrong about that.
Thanks. Olga, please.
Yeah, well, first of all, thank you for that lovely talk. That was really brilliant. I wonder how much is actually due to the fact that, due to the way in which experimental and empirical work actually functions in the system. Because there's a history always, and there's an initial question, there's an observation campus in the 70s when grid cells and actually place cells were found first. I wonder whether revisiting all the results and reintegrating them would be the way forward, because often, obviously, the results push you in one direction and create conflicts that are maybe not present if you think more about the functional context at a general level. What do you think could be done in that? I mean, I'm sure.
I'll tell you what I understood. So I think I kind of interpret the first part of your question along some mental model that I have. So tell me if I got it right. Is that perhaps this theoretical idea that we had building on the evidence so far can be revised moving forward, so we shouldn't basically, should we put it on hold or don't wait till we really think about, so is that yeah. No, okay, yeah, I saw they got
yes,
it wrong.
that's certainly along the lines. I mean,
Okay.
one of the major problems is when people revisit a particular area and discover new phenomena or whatever they want to explain then. The problem is that very often the tendency is to forget about everything that anyone else told you about that area
Right.
and found earlier and say, this is the function. And it gets really, depending upon what venue you publish and whatever, it becomes a dominant sort of way to discuss that particular aspect. And that can actually be in the way of making progress because you just, stuck with your own.
So short answer, completely agree. Longer answer, I think. So one is that as evidence is piling up, yes, the cognitive maps itself, as a model, is under a lot of pressure, even for spatial navigation. I think you know it much more better than I do. So some of the stuff that we have investigated in the lab is this phenomena called goal over representation, which is basically, okay, you take place cells to be firing in some kind of an objective way. They encode a single location in space, but if you insert a certain amount of reward, they start basically to attract to the location of the reward, to the location of the reward. So basically, is it an allocentric representation or something much more behaviorally weighted than we would like to think? I think that's correct. Look, there is always this kind of tension between, is the cognitive map the leading paradigm or is it a dogma? And I think this kind of speaks a lot to what you are saying. And okay, so now comes the tension where philosophers start to talk about what neuroscientists are doing. We will go like to the most finished science. There are a lot of reasons for that. We want the stuff that is much more established. I will go to the stuff that got a Nobel Prize because it has some consensus according. Although there might be a lot of recent results that basically contradict whatever like the cognitive map is saying. So there is this kind of, okay, in order to make a theoretical point, even like a conceptual one, you really need to go to the mature science. But is the mature science really a paradigm or a dogma? That's always, I think, is a tension. I think the cognitive map still has a lot going for it. I'm the most far from a skeptic about this, but yeah, completely agree.
Thanks.
Thank you very much for an exciting talk. Yeah, I would also support the view of my predecessor here. From an experiential point of view, you always need to consider these concepts in a historical perspective, keeping in mind what methods and what lines of empirical research were available to the people who developed the concepts. It's like in real life, if you have a hammer, you see a lot of nails everywhere. This is the way how empirical people think and talk about what they encounter. Nevertheless, I would start to reframe what you presented here, not as a kind of dilemma, but as a very exciting time now, because we can restart conceptualizing and rethinking problems that were typically separated, like episodic memory and spatial cognition. And now we have the tools and the methods available to combine them, to really understand what the hippocampus might be doing. And even saying that what the hippocampus is doing is huge simplification, because we know a lot about the hippocampus, how it's connected, how its subfields contribute to computations that the hippocampus produces in the output. And now we have the means available to combine them, to rethink this computational part, because we have the methods now to look deeper. We have the computational methods, we have the empirical methods, and we have the cross-species methods to look across all these parts. So from my perspective, it's not a dilemma, it's a great opportunity. And maybe here, I don't know how you think about it, but this work of Tim Behrens and others with the Trollmann-Eichenbaum machine is a very good example how you can also mechanistically, at least in computational terms, combine these two views very fruitfully to come up with results that were not available before. So I don't know what your take on it is, maybe a kind of Bouchaki's view, not to think about cognition and the brain from a human perspective with episodic memory, semantic memory, decision-making, perception, and whatever, but rather to use this opportunity to think the brain from the inside out might be a way to go forward.
So I want to say something about the historical point, because there's like two ways of looking at how the historical story of cognitive map developed. One is to, okay, they basically construct some sort of a moat, and we are now trying basically to see if there's a way to overcome what our predecessors have gave us. But another thing is that I think cognitive map has this distinctive feature, which is like one of the first constructs that allowed psychologists to escape the behaviorist trap. So this is like Tolman and like how can we explain more flexible behavior. It's not that cognitive maps explain all forms of navigation. It explains some sorts of the navigational tasks. And also, a lot of, so this ties back to like, do we need like these kind of sorts of cognitive structures that we cannot get if we don't think about it in that way? That doesn't undermine your point about can we keep on developing this kind of stuff moving forward. I completely agree that this is not a dilemma in the sense that we are stuck. That's what I was trying to refer in the end by saying it's not a stalemate, it's an embarrassment of riches. This is really a very exciting time. But my point was, by framing it as a dilemma, was to say that even if it's a very exciting time, it's not that like a time where anything goes. A lot of the historical trajectories and the finding that people have basically discovered all these kind of components that basically make the cognitive map to a very coherent construct, constrain a lot of the theoretical routes that you can take. This is why, like, Behrens appeals to, he calls it the Tolman-Eichenbaum machine. This is like basically, so even by itself, they are talking about spatial structures. And I think that these abstractions, like those state transition models, they really have a problem of coming back to physical navigation. We can talk about it later so it's very good to explain the memory stuff, but when you try to bring it back to explaining performance of rodents, it doesn't always capture the fine-grained details. Yeah, but we can basically, this is like a big discussion. Yeah.
Thanks, Jonathan. So I have one more question, but I want to keep Beate happy, so I'll ask you over coffee. Thank you very much.
Okay, let's start with our next talk. There will be a long lunch break. You have time for discussion then. Okay, so our next talk will be given by Anastasia Garballo. She is a PhD student at the University of Valencia. And she just told me that she changed her title a little bit, but she will explain why. So her title now is Where Representation Begins from Perceptual Constancies to Associative Learning. The stage is yours.
Thank you for the presentation, and hello, everybody. Well, in general, I'm interested in the question of where representation begins. So I started working on it, mostly reading these accounts about where the minimal condition that an organism has to fulfill in order to possess representations. But in the last few months, and I changed a little bit the working method, to say in some way, and I'm trying to focus in different organisms that has been postulated as minimally representational to see the reasons why they have been postulated as minimally representational, and to see a little bit which are the mechanisms or the biological grounding surrounding or that supports representation, and to see what's going on, if we can find a biological tipping point or something like that. So in this talk, I will focus on boss jellyfish because of that reason. It has been postulated as minimally representational. And I changed a little bit the name of the talk because of the experimental data. That's more, at least their most recent experiment have been focused on associative learning rather than in memory. So I thought it was more interest to do it like that. So to start with the talk, I will explore boss jellyfish as a minimal case of representations to find out which are the biological mechanisms that support these representations. So I will first discuss this claim that was postulated by Arnelos and Moreno in his paper of 2021. And they claim that the simplest organisms are boss jellyfish Tripedia cystophora because they do possess a size constancy mechanism. So in contrast to his proposal, I will claim that to possess representations, it's not enough to possess a constancy mechanism, but we need behavioral flexibility. So I will explore what this means in the relevant sense, and I suggest associative learning as a clear case of such a behavioral flexibility. So finally, I will agree with the authors in the claim that jellyfish are representational, but for different reasons. So the talk will be like that. First, presenting the problem and the theoretical framework, then their claim, and finally my suggestion of behavioral flexibility in the relevant sense, associative learning, and I will support my thesis in an experiment that shows associative learning in boss jellyfish. So about the problem, I'm working again in the question of where representation begins. So in this talk, I will try to specify if boss jellyfish is really a minimal representational system or not. And to do so, I will adopt an explanatory notion of representations. I think it's the most common nowadays, which says that representational states are legitimate if and only if they play a explanatory role in genuine representational explanations. And also in literature, we can find many different accounts of representation, and in this case, Arnelos and Moreno, they try to specify the minimal conditions for representations, but it do so by examining a specific organism that, according to them, is the minimal organism that fulfills these conditions. And according to them, this organism is boss jellyfish. So they examine or they focus in these organisms trying to specify if these organisms do fulfill the necessary conditions to ascribe them representations. So, again, Arnelos and Moreno have recently put forward an answer to this question, to a question of where representation begins, claiming that the simplest organisms that possess representation are boss jellyfish. Well, they explain how the visual system employs a size constancy mechanism that gives obstacle avoidance, and the use of such a constancy mechanism is supposed to distinguish them from inflexible organisms that has been defined as non-representational. And, well, I will first explain the contrast that they do between flexible and inflexible organisms, and then I will explain the free conditions that are supposed to capture this contrast and why they claim that boss jellyfish do fulfill these free conditions in virtue of possessing a simple constancy mechanism. And to do so, I will explain you a little bit how does this constancy mechanism works. And later I will explain my proposal that boss jellyfish are a clear case of representational system, but because they possess associative learning. So they claim that the employment of a constancy mechanism distinguishes boss jellyfish from inflexible organisms, like, for example, hydrozoan, which are categorized as non-representational. And they explain this contrast focusing on the behavior of both organisms. In the case of hydrozoan, its behavior is supposed to be triggered by a specific vibration in the water, so its responses to the environment are directly tied to a specific proximal stimulus without employing a constancy mechanism. And so, according to Arnelos and Moreno, their sensory states are mere indicators rather than representation, as they are only capable of direct causation and so inflexible behaviors. And in contrast to that, boss jellyfish is supposed to have flexible behaviors because in obstacle avoidance, boss jellyfish use a constancy mechanism that integrates different sensory input with the animal's perspectival aspect. And so, according to Arnelos and Moreno, boss jellyfish exhibit context-dependent behavior, and so the behavioral flexibility that is characteristic of representational systems. So, according to Arnelos and Moreno, this contrast between flexible and inflexible organisms are supposed to be captured by the following three criteria. That is, an organism should fulfill them in order to qualify as representational. First, they should cause successful action. Second, be decoupled from the structure of related incoming physical input. And finally, they should have exploitable structural similarity between the neural structures and the environmental features. And Arnelos and Moreno explains that boss jellyfish do fulfill these three conditions because these three claims. First, their distal action are based on neural structure, and this means that they play an essential role in obstacle avoidance. Second, they make use of a primitive constancy mechanism, and so the behavior is not determined by causal coupling. And finally, successful distantial action requires the capacity to construct neural structures that are structurally similar to environmental conditions. And that's how Arnelos and Moreno claim that neural structures in cubozoas or in jellyfish fulfill these three criteria, and so they qualify as representational. But now I will argue that this constancy mechanism is not enough to ensure the behavioral flexibility that is supposed to qualify an organism as representational. And to support this, I will show why the constancy mechanism of the jellyfish does not provide this behavioral flexibility in the relevant sense. So, a little bit about the boss jellyfish to know how does it work. In a simple way, the boss jellyfish perceive the size of the obstacles through visual detection using specialized eyes, and the size has two different filters that helps to determine the object's location. First, the contrast-dependent filters that indicate the object size based on contrast, shapes, and these kind of things. And second, the pattern-dependent filter that includes the animal perspectival aspect, so it indicates the distance to the object. And in this way, this constancy mechanism helps to locate objects by integrating sensory information about size and distance, and this integration is what triggers the behavior. So, well, to be more precise about the constancy mechanism, it works as follows. First, it processes visual input, and then the jellyfish eyes produce a signal dependent on the contrast and the size of the object. Then this signal is combined with other inputs to prioritize responses according to the most important factors, that size and distance. And then the jellyfish bell contrast as a result of this integrated signal, enabling movement to avoid obstacles. So this constancy mechanism, I mean the next one. Yeah, so this constancy mechanism meets the three criteria that I have explained above, because the size constancy mechanism gives actions allowing successful and adaptive navigation. Then the internal processing and integration of visual input is decoupled from the raw sensory input. And finally, the neural structure and environmental elements are structurally similar, providing a foundation for precise and context-dependent behavior. So the use of a size constancy mechanism that integrates proximal retinal stimulation with perspectival aspect introduce a level of decoupling from the raw sensory input, and so this sophisticated mechanism allows jellyfish to maintain unstable perceptions to an appropriate responses to environment despite the variation in the sensory input caused by change in distance or environmental conditions. However, I will argue that this constancy is not enough to qualify as representational because it doesn't provide the behavioral flexibility that is characteristic of representational systems, but I will finally agree with them in the claim that both jellyfish do qualify as representational, so we will see how. So first, I claim that constancy mechanisms are not enough for an organism to qualify as representational because these constancy mechanisms provide a form of sensory integration to guide behavior, but it does not allow for behavioral flexibility in the relevant sense. So the size constancy mechanisms allow jellyfish to integrate information from different sensors, that the size and the distance, and the combination of this size and distance information can indicate the same object in different situations. So they provide a stable perception, and in this way, the constancy mechanisms enable jellyfish to react in the same way to retinal stimuli of very different types. However, this capacity for constancy does not provide behavioral flexibility in the relevant sense because the relevant flexibility refers not only to the ability to respond appropriately to different stimuli, but also to the ability to generate different responses to the same representations. And, so this constancy mechanism by itself does not enable jellyfish to react differently to retinal stimuli of one and the same type, and so jellyfish behavior remain rigid and predictable in face of the similar representation if we only consider this constancy mechanism. So, in general, it is accepted that behavioral flexibility is the hallmark of representational system, but we have to be clear what does really behavioral flexibility means in the relevant sense. And as I have stated, behavioral flexibility in the relevant sense refers to the capacity of an organism to trigger different responses, not only to different stimuli, but also to the same representation. And this notion can be found in many theories of intentional representations that, in general, they claim that a representation must be able to guide behaviors in an adaptative way across diverse situations, which implies that the same set of representation can be processed in different ways depending on the context or the internal state of the organism. Or to say it in another way, the behavior of an organism that possesses representation should not be rigid, but it should be flexible and adaptative, as it can vary according to context and internal state. So behavioral flexibility indicates that the organism is not responding in a fixed way to the information it receives, and it shows that the organism is able to process representation of a situation or of a stimulus in different ways, and thus that the behavior can be dynamically adjusted. Well, now in this talk I will suggest that a cognitive or biological mechanism that ensure this behavioral flexibility can be learning, because it allows the organism to adjust its responses to stimuli depending on past experiences, and so it will qualify as representational. I claim that because through association, an organism associates a specific stimuli with a specific responses, and so it can modify its behavior based on past learning of past experiences. And so associative learning ensures that every time a stimulus is perceived, the organism can trigger different responses based on its previous experiences, current context, or its internal state. So associative learning implies that the organism is not responding automatically in a rigid or fixed way to stimulus, but in contrast, it shows that the organism can adapt and modify its behavior to the same representation. So when an organism possesses associative learning, it qualifies as representational because it's able to use representations that not simply reflection of the stimuli, but that give adaptative and variable behavior or context-dependent behavior, and so these behavior are based on representations. So the main question should be now is, was jellyfish to have associative learning? And a recent experiment by Bielecki et al. Shows that yes. Well, in the experiment, the boss jellyfish were confronted with obstacles of different contrast. And these different contrasts mean that they were more or less similar to the surrounding water. In the case of high contrast obstacles, that are very dark objects also, and so they are very easy to identify, the jellyfish automatically avoided them from the beginning with almost no collision, and over time the frequency of successful avoidance didn't change significantly, and this indicates that avoiding high contrast obstacles is happening without learning. But in the case of low contrast obstacles that are more difficult to detect, the jellyfish exhibited associative learning. Initially, they frequently collided with these obstacles, failing to identify them as dangerous. But as the collisions occur, they learn to adjust their perception of danger, associating low contrast obstacles with negative consequences that are the collisions. And finally, the jellyfish avoids the obstacles without colliding. Here you can see the more specific experimental results, only of the low contrast obstacles. I didn't bring the experimental data of high contrast obstacles because the change was no significance, so you have to trust me in that. And so, this experimental result shows that jellyfish are not born, to say in some way, with an automatic response, avoidance response to low contrast obstacles, but that they develop these avoidance strategies based on past experiences. So this qualifies as associative learning because jellyfish modify its behavior based on the relation between visual stimuli, the low contrast obstacles, and mechanical stimuli, that are the collisions. So they learn from negative past experiences and they modify its behavior accordingly. So to answer the question if jellyfish are really a minimal representational system, I say that yes, they are representational because according to the relevant notion of behavioral flexibility, both jellyfish do qualify as representational system as they do possess associative learning abilities. To explain this a little bit, if we accept that a fundamental characteristic of representational system is the behavioral flexibility, this means that an organism has to be able to generate different responses to the same representation. And associative learning in both jellyfish do fulfill this requirement because through association, jellyfish modify its behavior to representation of similar objects based on past experiences. And in this way, they do not merely respond or react to sensory stimulation in a fixed manner, but the responses vary dependent on past experiences, so they exhibit more adaptive behavior guided by representations. And I claim that this is a clear example of behavioral flexibility in the relevant sense, so both jellyfish are a clear example of representational system. And well, to conclude, my argument has been the following. According to Arnelos and Moreno, both jellyfish qualify as representational because they use a safe constancy mechanism. However, this constancy mechanism is not enough because it does not guarantee behavioral flexibility in the relevant sense. And for an organism to qualify as representational, what we need is precisely this behavioral flexibility, but understood as the capacity to trigger different responses to the same representation. And a clear example of it can be associative learning, as it enables to modify the responses of the organism, ensuring that the organism is not merely reacting in a fixed manner to the stimuli. And recent experiments have shown that associative learning abilities are present in both jellyfish. So both jellyfish do qualify as representational organisms because their ability to learn associatively provides them with the behavioral flexibility that is necessary. I want to clarify, just in case, that even if I'm interested in the question of where representation begins, I'm not claiming that associative learning is the necessary or sufficient condition for having representation. What I did is to focus in an organism that has been postulated as minimal representational, and what I conclude is that it's more complex than when we thought 3 years ago, so it's not a minimal case of representations, but a clear case of them. And that's all.
Thank you very much for this interesting talk. We are waiting for the microphone, so I will keep talking here. We have plenty of time for questions. So I saw Adrian first.
Yeah, thank you, Anastasia, for this clear and interesting talk. I have a possible objection. The objection would be that you might conflate two different norms. So the representational norm is accuracy, so an organism represents how the world is, and the other is practical normativity, evolutionary normativity, what the organism is supposed to do with that fact. And the question is, I might be a representational system, a genuine one, just to know, for example, where the coffee is, but I might be very inflexible concerning the coffee. I always go to the coffee, but I still know where it is. So in your account, if I understood it correctly, I would not count as a representational system because I don't have a flexibility concerning the whereabouts about the coffee. What would you say to this?
But what will you do? I mean, you know where the coffee is, and if you really have a representation of it, you could go to search it, you could drink it, you could avoid it, or I think I'm not taking the point of the question, sorry.
Well, I still know it, right? I still know where the coffee is, or maybe I know where my home is, for example, but I still have one way. I'm not very inflexible of getting home, especially today, concerning the strike, yeah. But I still
Okay.
know it, right? So I would be a representational system.
So the point is, knowing where an obstacle is does provide you with representations.
Yes.
Well, I don't think so, because most of the complex or even not complex organisms have sensory input or one way or another to know about their environment. And I think that's not enough to possess representations. I mean, the plants also react to the environment. So we could also explain a plant's behavior as it's moving because the plant knows where the sun is.
Well, yeah, I'm using the sense of knowing in the perceptual constancy sense. So I'm, yeah, but I don't want to monopolize this. So.
Yeah, thanks very much. Great talk. I like that a lot. Just a mechanistic question, really. How do they get the distance information? Is that stereo vision, or how do they get distance information? You said it's important that they know about their size and the distance to the objects.
O sea, that was a question about how they perceive.
Where do they get the information
Okay.
on distance from? Yeah.
In a more biological sense. The jellyfish has a rophalium that is kind of the most central or important part of the nervous system, where most of the neurons are. And in this rophalium, they have 4 eyes. 2 of them serve to detect the environment. So using them, they can perceive the contrast, the shapes, and so on. So they know the size of the object, and then this information goes, it's mixed in a pacemaker with the animal's perspectival aspect, and then the signal is triggered. Some say that both jellyfish, in this case, because they are the most complex jellyfish, they have a kind of centralized nervous systems. Others, they say that it's not enough to qualify as a centralized, but they have a rophalium where the lower lens eye are with other eyes, and this is the important part for the sensory stimulation. I think it was the question.
Yeah, thanks, that was really interesting. So I was wondering about how to understand this idea of different responses to the same input representation, like what's underlying behavioral flexibility. So perhaps I misunderstood the experiment. So do they keep having 2 different responses to the obstacle, or did they at T1 have 1 response, then they learned, and now they have another response, but it's basically just 1 response that have changed over time?
Yeah.
Is the second one the latter or the former?
It's the second one,
Okay.
but it exhibits a more clear case of behavioral flexibility and representations because, can I move a little bit?
You might need a microphone when you keep talking.
If we only use the constancy mechanism, it will be like the size and the distance are perceived, but different combinations of those informations can indicate the same object size or the same object in general. So the representations that it's in the constancy mechanism, the integrated information that should be the representation will trigger always the same behavior, because what is invariance is this part and not this one. So in the second case, the relevant part is that representation can trigger different behaviors. I mean, if we have one and the same obstacle in different, more or less proximal, it will always respond in the same way, like colliding to it, but with the associative learning, the same representation of one object or similar object, because they were different objects or low contrast, they trigger different behavior, because now they are not only able to collide with them, but also to avoid them. It's true that being what they perceive is mostly contrast, shape, and these kind of things. So the a low contrast obstacle can be, well, I think it could be, it's also related to the case of misrepresentation, because when a low contrast obstacle is perceived because of the surrounding water and everything, it can be engañoso, like they can perceive it in a bad way, or yeah, like a mistake, in a mistaken way. So maybe in one case, as they are misrepresenting it, they can collide with it, or the avoidance can be not very clear and almost colliding, or it can avoid it in a very long distance, so very successful action. And this cannot happen only with a constancy mechanism, because if the response is directly tied to the proximal stimuli, it will always react in the same way. So this schema was a little bit to see the difference, but here we don't have this step. I put it to have it more clear. But if it's fixed, it cannot behave in different ways with the same, to the same obstacles. So
B2 comes after B1.
what?
It comes after B1, no?
Maybe.
Because they need to first have B1 or something like that?
Yeah, they modify the internal states, the nervous system, and so on. So now, after one representation that it's the obstacle, can choose between different behavioral options.
So it's a switch, basically. You have either B1 or B2.
Okay, so you won't be hearable on the stream, so we just proceed with our list of questions, and maybe you can have one further question afterwards, and you get the microphone. So Marcus is next.
Yeah, thank you for a very convincing and well-structured presentation. I'm not a philosopher, so maybe I'm missing the point here, and this is probably more kind of a clarification question here. I do not really see why behavioral flexibility is a necessary condition to be a representational system. For example, I can have a pocket calculator, and if it has some input, some sensors, I can put 5 plus 5, and it certainly has an internal representation, because in its registers and buffers, there is a representation of the 5, of the plus sign, and of the 5, and it produces an output, which will be 10. And every time I come to the representation of 5 plus 5, the internal representation, even via different ways, it will hopefully always tell me that the result is 10. So it certainly has an internal representation in this case. So for me, this would be a representational system. Behavioral flexibility, on the other hand, relates to adaptivity. I can be behavioral flexible in terms of being adaptive. But I don't know why this is I don't see immediately why this is a necessary condition for a representational system.
Yeah, I think maybe that's a debate between liberal and restrictive representational accounts. Many of the liberal accounts, I think they will accept what you are saying, that that's a case of internal representations. But in my case, I'm more restrictive. And for example, well, so when the organism do receive the 5+5, it is receiving different sensory input and they are combining them, so it will be the first case. So it's creating it's integrating different sensory input, it's triggering an output, but I think in that case, representations didn't play any explanatory value because we could explain that case only saying that the response is directly or automatically triggered by the proximal stimulus, or in this case, the 5+5. And so for me, this is not a case of representations because it cannot vary the response in normal as you say, it will always tell you that the response is 10. So if the response cannot vary, this means that the physical input are creating automatically or in a fixed way the response, and so for a more restrictive account of representations are not playing any explanatory value there. So I don't know if it answers you a little bit.
Yeah, we have a coffee break and a long lunch break. You can keep on discussing that.
Thank you very much. A comment and a question. So the comment, I think, is pointed back at Adrian. I think your account does have a way to block it from him going to the coffee all the time, right? Because you said that the representation is also dependent on the internal state of the system, for instance. And if he keeps going to the coffee all the time, eventually he would bust, right? So he better not drink coffee all the time because in the end, I think there would be a cost associated. A question for you, because there are some anti-representationalists in the audience. I'm on your side, but maybe I want you to say something more about why we need to worry about jellyfishes being representational systems versus just being, cognitive systems, and what you require for representation here is something like associative learning, which gives behavioral flexibility. Why not just say, look, I'm happy to accept that jellyfishes are cognitive systems, but the reason they are cognitive is because they have different mechanisms and also associative learning capacities, and then we don't actually need representations.
In the first case, just to point out, maybe I didn't get your point, it could be a case. When I'm saying that a representation triggers different behaviors depending on the internal state, what I mean, it's like, for example, if you are hungry, you will eat, and if you are not, you won't eat, even if you have the food there. So the internal state, the variation of the internal state refers a little bit to these cases that are very typical in the literature. And to the second point, why should we ascribe representation to an organism? For me, it has been, since the first time I heard about representations, it was very obvious that we need them. So most of the time, I suppose, or I accept that they exist, but I think they can play an explanatory value, and I think that's the main reason by which we accept representations. In the case of boss jellyfish, well, I bring them, they are important for us because they have been postulated as the minimal. So to know which are the biological grounding of representation, we should examine these borderline cases. But why should still ascribe representations to them? Because they show a really complex behavior. I think that representations make mostly sense when we have a neural system. So, because when we have a neural system, some content can be fixed in this, well, it's fixed in some, to say in somehow, in these neurons, so, or a group of neurons that can modify the behavior, make it more adaptive, and because without this behavioral flexibility, these changes in the behavior, in the internal states or internal organization, we couldn't have explained the evolution of many organisms. So, and I think that's the explanatory value of representations. I don't know, I'm not interested in the debate between anti-representational and representational organisms, so sorry. They exist and that's all.
Yeah, I guess I stay with that question of how do you define that. And it took me a long time to actually get to a point now where I can actually accept the fact that it makes perfect sense to use that distinction, basically, of a representation. And that is simply because a lot of connections between sensor input and motor output, they're immediate, and you really don't think about representation. Think about your spinal reflexes controlling your leg movements. There is a sensor that measures information in the exact same coordinate system as the muscle system or musculoskeletal system that's controlling the movement. You don't think about representation here. You could say, okay, I can quantify that mathematically, and then I have a variable, and then I can say, this represents the length change of the muscle or something. But that doesn't really make sense from the functional point of view. But if there is some information that you need to store, because you later on a different functional context, you have to resort on it and recall that, that is when actually it makes sense to think about representation. And that's actually where, in the end, if you think of forward models or inference copies, something like that, which are a part of stored information, then you can really think about representation and use it. I think that's important. In a way, the flexibility, I understand why learning and flexibility in the end kick in that system as well. Because obviously there are conditions under which the relationship that was established in that animal breaks. Because if you change the experimental conditions, and it starts colliding. Mm-hmm. Now then the representation needs to be updated. And that means representation and updating mechanisms, in a way, learning mechanisms associated or whatever you want to call it, are intimately linked somehow. So that is another combination of features that are important, I suppose. And it's really interesting. I mean, from that perspective, yes, you have a representational system there.
Yeah, even in the paper of Arnelos and Moreno, even if I don't agree with them, they talk about this feedback and constant, this feedback between the input, the internal state, and the output, and so that's the point. About the first part, I think it's a key point what you say about the storing information, and that's why my first title of the talk was memory, but I think I need to learn more about memory in order to be able to talk about it, so, but yeah, I think the point is by there.
Okay, so let's thank Anastasia again.
Thank you.
I suggest we have a 15-minute break now, so many questions.
Okay, yeah, we will be continuing. Our next speaker is Matt Kohar, who is also part of the NeuroExp project. He is working on mechanistic explanations and more broadly also on the philosophy of cognitive science. He has published a great number of papers and also a book on that is called Neural Machines, and that's has come out 2 years ago, I guess. Yep. Yeah, 2 years ago. We're very happy that he will give a talk today about lessons from Locust's individuative role of representational content.
Okay, thanks, Peter. So this talk is going to be about the Locust case that already Holger explained yesterday, and now he explained it well, and I'm going to probably explain it very poorly, okay? But we'll see what happens. We'll see how it goes. And this is one of the key case studies in our project, so this is why I'm working on it. Okay, so let's start with looking at the phenomenon of locust threat detection. So, does it work? It does. There we go. So, the phenomenon is, I take it basically this: when locusts are confronted with flying objects, with objects that are looming, they jump and then they fly away, so they don't get eaten. That's the idea, or that they don't get like smashed into a wall or something. Okay. And so, oops, that was back. Good stuff. So this behavior appears to be sensitive to the angular speed of the approaching object and to its angular size in the locust visual field. So this is what it looks like. There is the looming object, which is just a square that moves towards the locust's eye, and you can see that it subtends a certain visual angle in the locust visual field, and as the object kind of moves towards the locust, that angle itself increases. That's going to be, the rate of that increase is that angular velocity. And of course, there is then a sensor that also senses the instantaneous angular size of that object. Okay. Oh, that was, I keep pushing the back button for some reason. Okay. And so what is the explanation for this behavior, or at least part of this explanation? Well, part of this explanation is, to a first approximation, neural computation that is done in this single neuron. So there is a neuron, which is the lobular giant motion detector, LGMD. This neuron receives inputs correlated with size and speed of expansion and commands a jump when the object appears large enough in the locust visual field. And the activity of the LGMD correlates to the product of angular speed and angular velocity of the approaching object. So, one way to cash that out would be to say, okay, the LGMD then computes the optimal time to jump by multiplying angular size and angular velocity. So, in a paper by Jones and Gabbiani, you can see this nice figure which shows you the inputs received by the neuron correlated to angular velocity here, to angular size here. These inputs get compressed into a logarithmic scale, then added together, then re-exponentiated, And if you do that, you get basically a multiplication of those two inputs. And that then determines the firing rate of this neuron. Oh, again, the back button. It's just too far. Okay. So, what I'm going to talk to you about today then is how this relates to individuative role of content for physical computation. So I'll look at the account by Oran Shagrir. Now, I think this case study shows that there are some complications to this account. And so I will tell you about those, and then I will just conclude. Okay, so it's going to be fairly straightforward, hopefully. Okay, so what do I mean when I say individuative role of content? Well, this is part of Shagrir's semantic view of computation. So according to Shagrir, if we want to ascribe computation to a physical system, we need to also ascribe some representational contents to the states of this system. So in other words, no computation without representation. But also, the idea here is that semantic contents help group physical state types into computational vehicle types. So we basically figure out what groupings of physical states are computationally relevant by looking at their representational content. And the main argument that Shagrir gives for this view is from this kind of tri-stable physical gate. So imagine you have a gate, you have like a physical system that has two inputs and one output, and the inputs and outputs are both just voltages, and there are kind of three stable states that each of them can be in, either high voltage, medium voltage, or low voltage. The exact values don't matter really for this. So Shagrir points out that with this system, depending on how you kind of group those physical states into computational vehicles, you can implement two different types of logical gate. You can implement the AND gate. So here is the logical form of the AND gate, 1, 1, 1, 1, 0, 0. We know what an AND gate looks like. And now Shagrir points out, look, if you take this tri-stable gate and say that only the high voltage correlates to 1 or true, and both the lower voltages correlate to 0, you get an AND gate from this. So the first row of our kind of logical form corresponds to just the first row of our physical description. Then the second row will correspond to those two kinds of physical states. Then the third one will correspond to these physical states, and the fourth one will correspond to these physical states. So far so good. But of course, the problem is you can also take this exact same physical system, I didn't cheat, the figure on the right is still the same, and implement an OR gate with this. And this happens if you group together the high and the medium voltage into one type of computational vehicle. So if you do that, then the first row of our logical description corresponds to these rows of our physical description, the second to these ones, the third to these ones, and the last one is just what's left. Okay, and the idea now is, Shagrir says, we need to take into account the content of these physical states to determine which of these kind of groupings we should choose, right? So if low and medium voltage have the same content, then we should lump them together and say that this is an AND gate. If medium and high voltage have the same content, we should lump them together and say that this is an OR gate. Okay? How does this relate to the locust example? Well, in the locust example, we also have kind of multiple potential vehicle types. if we just, pre-theoretically look at the system. It could be that the system computes with spike timings. It could be that it computes with instantaneous firing rates. It could be that it computes with average firing rates and many more others, right? And so the idea here can also be that looking at the content, representation content of each of these potential vehicles is going to tell us which of them we should choose. So one option would be to say that the fact that the timing of highest spike frequency correlates to the locus jumping selects it as the correct output vehicle here. Okay. Next point, or next part of Chagrir's account is what he says about how we determine whether a system computes or not. Right? So this is very formal and very complex. So I made the drawing for you. Okay. So here we have three systems. So here is our physical system, which has inputs and outputs and performs some physical transformation on this, some transformation G. Right? Here is a target domain which just contains some external objects or external properties and some relations. And Chagrir's idea is the first condition in order to say that the system P computes is that it should mirror some relation in this target domain. Okay. The second condition is that it should implement some formalism, some formal transformation. Implementation in this sense is just a mirroring relation as well. So we have this kind of double mirroring where this system G at the same time implements this formal system, but also its transformation mirror the relation in the target domain. So if we put in X and we take out Y, that means or that can tell us that X with an underscore stands in relation R to Y. And if we have this double mirroring and at the same time inputs to this physical system and outputs from this physical system actually represent these elements in our target domain, then we say that the system computes. So here is an example from Chagrir, a neural integrator which Chagrir says computes I positions from I velocities. So it integrates over I positions to get I velocities. We can describe the physical evolution of this neural system by saying that it integrates its physical inputs, but also the relation between I velocities and I positions, it's itself an integral relation. And as it happens, the inputs to this integrator actually represent I velocities and the outputs represent I positions. So this, according to Chagrir, justifies the claim that this is a system that computes integration. Okay, how much time do I have? I never know if I go too fast or too slow. It's awful.
We're talking for 16 minutes.
Okay, cool. So, what are some issues with this? So let's take seriously this idea of mirroring, and let's say that our computation, our neural computation, should be described in a semantic manner, right? well, then we need to say, what physical property is being computed from which inputs. And in the case of the LGMD, one option is to say, so this is the input side. According to Jones and Gabbiani, the LGMD multiplies angular velocity with angular size. Okay? Now, according to Gabbiani et al. 2002, the LGMD computes an angular threshold. Right? So in our target domain, we should have then these three properties related by this formalism of multiplication. So angular velocity multiplied by angular size, we compute an angular threshold. So here is what Gabbiani et al. Asked themselves, how does the LGMD compute this angular threshold? And then they explain that it multiplies things together. And the idea is, if the approaching stimulus achieves a fixed angular size theta at time t, then the LGMD firing rate will peak at some time t plus delta. Okay, oops. And I don't know how big this is, but you can see that in here on these graphs here. So the idea is, for each firing rate, there is a fixed angle that the stimulus will reach in the course of approach sometime delta after this firing rate is reached. So also for the peak rate, there is going to be a specific angle that the stimulus will reach after this peak rate was achieved. So one idea then is, okay, the LGMD computes this angle. But the problem is, if we're really saying that the LGMD multiplies angular velocity with angular size to compute angular size again, things don't work out so well for us. Because if we do some dimensional analysis on that, what we're saying is that we are multiplying something in radians per second with something in radians, and we're getting back out radians again. Right? So it just seems that angular threshold cannot be the product of angular size and angular velocity in our target domain. So this assignment of contents then is inconsistent with the idea of what computation is performed here. Okay, but maybe I'm getting things wrong here. Maybe what we mean is that the LGMD actually computes the time, right? So it doesn't compute the angle, maybe it computes the time at which this angle is reached. So again, our starting point is that the computation proposed is a multiplication of these two variables. And now, according to Papayanopoulos et al., who also analyzed this case study, the result of this computation is going to be the time to jump, or when the angular threshold is reached. So on this interpretation, the output vehicle is the timing of the peak firing rate. But again, we have a similar problem, namely that the result of multiplying angular velocity with angular size cannot be time. It's going to be something in radians squared over seconds. So that cannot, just cannot be time. The assignment of contents, again, is inconsistent with the computation performed. So what we can say, of course, is that the LGMD determines the time of the jump by firing in this specific way, but it doesn't compute it from the inputs, from the external inputs. Okay. So another option would be to look more closely at the kind of biomechanics of the jump. So when the LGMD firing rate, the LGMD firing rate actually correlates with the tension in both flexors and extensors in the locust's leg. So I think Holger also already explained that yesterday, right? That or I don't know, maybe he didn't. But basically, the locust has to jump in like a very spring-like manner. So when it's preparing to jump, it cramps all the muscles in the hind legs and then releases all the energy in like a spring, so that it can get a good launch. So as the LGMD activity increases, tension builds up in the muscles. Now, once the inhibitory input from the angular size to the LGMD overcomes the excitatory input, this tension is then released. So it looks like actually this kind of very steep fall in the LGMD firing rate is crucial to the phenomenon. It's crucial to the release and to the jump. This is at least how I understand it. I might be wrong. So, but because of this kind of correlation, there is then the question, does the LGMD compute muscle tension actually? So, as you might guess, the answer is no, it cannot be. So again, whatever is computed is supposed to be the product of angular velocity and angular size. And we again see that this is radians squared per second, or perhaps just frequency, because radians are sometimes viewed as dimensionless. But it's not muscle tension, where you would have to have newtons in there. So of course it is true, if I'm right about my interpretation, that the LGMD sets the muscle tension, but again it doesn't compute it. Okay, now how much time do I have?
Maybe 2 or 3 minutes.
Okay, should be doable. Good. So one final option is that maybe the LGMD computes its own firing rate. Right? So here is what Gabbiani et al. Say in 1999. So at the algorithmic level, we can break down the computation performed by LGMD into 4 distinct steps. A calculation of the angular velocity, a calculation of the size subtended by the object, a multiplication of these 2 positive variables, and finally, a transformation of the result into firing rate. So maybe this is going to be the firing rate that's computed here. And so this fact actually then accords well with the fact that the entire time course of the instantaneous firing rate of LGMD is determined by the function from angular size and angular velocity, not just the peak or timing or anything, the whole time course of that firing rate. And when Gabbiani et al. Described this model in an equation, you can actually see that this parameter here, or this thing that's being computed here, that's the instantaneous firing rate, right? But this is still going to be a problem for Chagrir's account, because on this view, the firing rate is somehow both the output vehicle of the computation, but also the result of the computation in this kind of semantic sense. And that seems like also it cannot be. Yeah. That seems like that's an alternative that is not contemplated by Chagrir's view. So now I conclude. So representation relations may wow, I need to show you what I'm saying. Yes. So representation relations may individuate output vehicles in ways that are incongruous with the proposed computational formalism. I think I showed sufficient examples of how that happens in this case. Describing a neural computation in semantic terms may be difficult in cases where the output is a motor command, and there may be no straightforward way of identifying which relation in the target domain is mirrored by the computing process. Thank you.
For this fascinating talk. So, yeah, let's start with David.
Great, thanks. That was great. It's interesting. I've never seen a units analysis that's done like that on a neural computation. But one of the things I was wondering is if, I'm wondering about the propriety of doing that. That's certainly a physics technique that you're importing in neurobio, and I'm just not sure that tracks. When I have my neurobio hat on, I'm like, wait a minute. What's going on here is some sort of purely causal process, and then maybe there's some other variables that look like the variables that are being named, described a certain way by the neurobiologist, but actually are other internal variables that the brain is computing in various ways and so forth. That's my first, there's a propriety concern I have, but then there's a second question here, which is maybe if we take on sort of the street cred of a units analysis imported from physics, and maybe like, okay, that's fine, you can do that, the math works out. And it looks like it is computing some sort of radians squared per second. Sounds a little bit like a acceleration, or I don't even know if what that is. I don't think that works either. It's some whatever unit it might be, okay? But maybe that's fine because in the ecological demands that are placed on the organism, computing that thing is close enough to the output variable that you want to call force or whatever it was, such that the system can use that computational transform in a way that's appropriate for the action that the animal needs to produce. So there's a propriety question, then there's this, well, maybe it is this other thing that's actually being computed, and it just so happens that the trace of that function is close enough to the thing that our theory says should be computed.
Yeah. Thanks, David, for the question. So just to be clear, yeah, I'm not saying that the fault here is with like Gabiani and Holga and everyone who's doing the model, right? I mean, maybe the issue is the following, right? We have this semantic view that Oron is proposing, right? And according to that view, there better be some relation that is semantically describable that mirrors that physical transform, right? Now, the neuroscientists do also describe this transform, at least on the input side and on the kind of transform side, as multiplying these quantities, right? But the problem is, according to Oron, there must be input-output mirroring between the target domain and the physical domain, right? And so the output, the semantic output, better be related to the semantic inputs in the kind of relation that the that the computational description says, right? So that I take is what Oron is committed to. In the neural integrator case, this works out very well, right? Because it is actually the case that integrating over velocities gives you positions, right? But if you say, well, semantically, I'm here calculating or computing, you know, time or angular size or whatever, then you cannot say, well, you know, actually, I'm calculating it from inputs that cannot, by the proposed transform, give you that output. Of course, it might be that there is a different kind of further transform that happens on this. But yeah, then we would need to like also hear more about it, right? So you're really pushing back against the semantic. Yeah. So yeah, I don't want to, you know, not saying shut down the locust lab or anything like that. That would be silly.
But first, it's Alexander. Yeah.
Yes, thank you for your talk. I'm very sympathetic to the kind of arguments you make. I wonder if they can also be, I mean, I'm kind of sure you would disagree with that, but I wonder how far they can be extended to generally be skeptical of computational formulations of this kind of dynamics that we see there between the input neurons. I'm just going to call them retinal neurons. I don't know what they're called in locusts, and the LGMD. Because what you described very well is actually that, you know, the dynamics of how the LGMD responds actually seem to be relevant for what's happening here. So it really is that the fact that it reaches some kind of threshold quickly and then drops off even more quicklier. And I'm wondering what you would say we are gaining from describing this interaction between retinal neurons and the LGMD in of computation, in terms of saying it computes by multiplying the angular velocity and the angular size of the approaching object, rather than just saying we're going to build a model which is based on functions that describe each neuron's activity patterns and how they are coupled with each other, and just say, well, you know, these functions are instantiated in the organism, and that allows it to jump away when something approaches. Thank you.
Yeah, thanks. So generally, I would say that, you know, these kind of computational models are predictably very successful. I would say even explanatorily successful, and you are going to like end up describing the interactions on the physical level, in the physical system, in mathematical terms generally, right? So if we want to call that neural computation, I'm on board with that. Actually, we can you know, say that. I'm even on board in taking it seriously. I think the issue is when we say, oh, we need to look at, you know, representational content to see if it like really computes. You know, I think this example shows that can go wrong. So what I want to push actually is, you know, let's forget about the representational contents. Let's focus on, describing the physical dynamics, as you say, and then, you know, let's call them computations. I'm fine with that.
Okay, Holger.
Yeah, thanks. That was interesting, a perspective I haven't thought of before, obviously. But anyway, I think the mathematical description that's brought there, that was used there, the purpose of that was actually to explain basically the behavior, the dynamic behavior of the cellular response in terms of the input to the system. And I don't think there's an inconsistency at that side. So I have to check the units again, but I don't think there is an inconsistencies of that. So that's working. I suppose the parameters that in the end are decisive to control the behavior is not, so to speak, a time-continuous function of the firing rate, but it is a non-linear sort of threshold operation that's based on the firing rate, can initiate different sequences in the behavior that follows, cocking the hind legs, preparing for jumping, and then eventually release that energy for the jump. So there is, so to speak, a disconnect in that sense between the description of the input-output operation in that neuron and what is ultimately controlled. That's a very, very different situation from other systems I was talking about. For instance, these optic flow neurons, where you have a time-continuous output, and that will very likely be used on a time-continuous basis to control flight behavior. Because in that case, you don't have a threshold operation. Well, it could still be somewhere down the way, but that's slightly different. But yeah, anyway, and then of course, it is, in this case, I always had, and we discussed that earlier as well, I had a little bit of difficulties trying to see whether the concept of representation in this particular case is useful. Well, if you say, of course, if you have, so to speak, even if you go for a mathematical description, you have your variables, and you can say those variables represent something, right? But it's from the functional perspective not necessary to discuss that part. It's more important to see, okay, how is the peak firing rate related to a certain angle on the retina, which is the behaviorally relevant parameter in the end.
So, to your first point, of course, yeah, we probably need to look even more into how the LGMD signal gets like transduced into motor outputs to see, where exactly the behavior relevant properties of the firing rate are, and I thank you for that comment. To your second point, I think here, for Chagrir, he specifically says that he's just going to be very pluralist about representational content and about, how to ascribe it. So if in this sense, if in this case it is, enough to say there is some correlational information, that would, for his purposes, to individuate the system as a computing system already suffice. So that's enough content for this purpose, but it might not be enough content for like other purposes, perhaps.
Asaf. Yeah, all right, thank you. We discussed this a lot, but something that just popped into my mind when you presented the presentation was, let's leave aside the Chagrir's account for a moment, but focus on the problem with the units, right? So I think that one of the, let's say, so the units are like angular size and angular velocity, and there is a computation over them. But when you talk about the neural inputs, then you don't need to correspond to the units anymore. So you can say that this is a representation of the units, but it's shown with, I don't know, firing rate or chemical response or something like that. So maybe you could say that these are representations and the computations are formed over them, and you don't need the units anymore, the physical units in the world. That's one of the purposes of a representation, I guess.
I would think, though, that in that case, if you adopt that position, then you don't actually need the representation relation anymore to individuate computation, right? Then you just say, here is a bunch of like physical transformations. They are all transformations on, I don't know, firing rates or on cell membrane potentials or what have you. You know, pick your vehicle. We'll call these computations, and we don't care about the units in the like outside world because it doesn't matter for our purposes. And the significance of the like missing units comes up. That's like specific to Chagrir's account, I take it, because as Holger also pointed out, yeah, the units in this equation, which is the actual model equation, do work out. Because what is the result of that equation is that instantaneous firing rate, and oops. That's 1/2. Yeah, that is 1/2, and because radians are dimensionless, technically you can just not care about the radians and you get 1/2. So in that sense, the units do work out. It's when you try to like look beyond the firing rate into, you know, what might be the representational content where this problem arises. It's going to be, you know, whatever the, I guess, so it depends on you know, what theory of content best serves your needs, but it could be things that are A, behaviorally relevant, and B, like well correlated with the properties of the firing rate. So the ones that I looked at would be those, that angular threshold, maybe the time of the angular threshold, yeah, in this case.
Also this analysis, I mean, we discussed the case a lot, but now this analysis that you've given, that there's kind of a disconnect between, on one hand, the formula, neuron depends on these two other parameters, and then kind of, yeah, this representational characterizations that are found, which say, well, okay, it's the threshold angle is reached if you look at the input side, or it's time to jump if you look at the output side. And in this respect, yeah, this case does not cohere well with Oron Chagrir's model of computation. But yeah, maybe yeah. Other cases like the B case, I was of course always also thinking about that in comparing. Then there might be some better match, but yeah. So thanks a lot. So
maybe
yeah.
that's why Oron left early. Maybe that's why Oron left early, right?
Ah. Yeah. Okay, great. So let's thank again Matt, our speaker. And yeah, now it's lunchtime, and we reconvene at 2:30 again. Is that right? 2:15. 2:15. Oh, 2:15. Okay.
So, hi. Our next speaker would be Beate Krickel. Beate is one of the PIs of our NeuroExp project. She is a professor of philosophy of cognition in TU, and she is also the dean of the Faculty of Humanities and Educational Sciences. She's currently mostly working on mechanistic explanations, especially in the cognitive sciences or new sciences. Apart from many articles, she has written the book, The Metaphysical Commitments of the New Mechanistic Approach, which on a personal note really helped me understand the mechanistic approach, so thank you. And she's gonna talk about integratable or incompatible different explanatory approaches in cognitive neuroscience. So.
Yes, thank you, Asaf. Okay, so what I'm going to talk about today is, actually something, partly something that I've been working on, I think 2 years ago, and I already apologized to some of you. Some of you in the audience might have heard the first half of the talk already, but the general idea is to apply this model that I developed 2 years ago to a new area. So this is why I have to introduce it again. And so this discussion is starting from the observation, which became already clear during this conference. There are different explanatory approaches in cognitive neuroscience. So on the one hand, there is the mechanistic explanatory approach. We have also topological explanation. We haven't heard, Alexander, I think, talked a bit about that. We have dynamical explanation. We have computational explanation, representational explanation, and many more dynamic explanation, and so on. So there are even more explanatory approaches than we addressed during that conference. Mechanistic representational computational explanation being at the core of our project. And now philosophers and scientists alike, they wonder, so how are they related? What is the relationship between these explanatory approaches? As you can see, I grouped some of those here. Not much depends on it. I just wanted this picture to not have too many arrows. So now there are different views in philosophy. Some say, well, actually there's just one proper explanatory approach, and somehow all the others can be reduced to that one. So the mechanists are sometimes read as making this claim. Some go pluralist and say, no, these are just very different approaches, and it's fine if they can't be integrated. We just go pluralist. And what I'm going to argue for, I think, is somewhere in between. So saying that, or I believe that we should say something about how these approaches can be integrated and somehow to get a holistic picture. And anyways, they are all explanations or in the area of explaining cognition. So somehow there must be something to be said about how these explanatory approaches relate. And today, because I can't talk about all of them, I of course talk about mechanistic explanation and then about topological explanation. And this is what, like the older account that some of you have heard about, at least might talk about. And the idea then is that to start with computational explanation and see whether this approach can be generalized and also be applied to computational explanation, and then in the end to representational explanation and maybe even others. So as Asaf said, so I've worked a lot on mechanistic explanation, so this is for me the starting point. So I want to first explain what mechanistic explanation is, and then start with topological explanation and tell you what I think how these two accounts or explanatory approaches can be integrated. Okay, so let's start with what is mechanistic explanation. Those of you who know that now can, I don't know, write emails and come back when I talk about computational explanation. Okay, so here's a very, very brief summary of what the so-called new mechanists think what mechanistic explanation is. As you can read in many publications by these new mechanists, a mechanistic explanation explains the behavior of a system in terms of the causal interactions of the system's parts and their temporal, spatial, and hierarchical organization. We don't have to go into much detail regarding all these different concepts. Of course, this was basically what my book was about. I have a chapter on all of these concepts, more or less, but here's an example. So here you see a behaving system. So it's a body, it's a human body flexing a muscle. And now part of this mechanistic explanation is this. So what you can see here is something like a zoom into the muscle, and you see different protein filaments, myosin and actin, and how they interact. And I mean, that's already a really fascinating mechanism, that the myosin somehow attaches to the actin and then moves it along and repeats this behavior such that the muscle shows this behavior. So, and now it is said, so on the right-hand side, you see at least part of the mechanism for this behavior of the muscle, and there is supposed to be an explanatory relation between this mechanism and the behavior you observe on the left-hand side. And now this explanation is supposed to depend on or be grounded in a part-whole relationship between the actin, the myosin, and their behavior, and the muscle's behavior, and some kind of dependency relation. So the actin and myosin are somehow part of the muscle, and their behavior happens at the same time as the muscle's behavior, but this is not sufficient. You also need some kind of dependence. So what the muscle is doing depends on what the actin and the myosin are doing. And this dependence relation is often called mechanistic constitution in the literature. And now to understand the mechanistic account and also to understand some problems that arise with the integration question, we need to understand a little bit more what mechanistic constitution is supposed to be, or you also sometimes, or it's actually more common that the mechanists call that constitutive relevance. So saying that what the actin and the myosin are doing is constitutively relevant for the muscle's behavior. And this is the case if the following holds, at least according to a very popular account. The mechanism for the phenomenon P consists of those and only those acting entities that are constitutively relevant for P. And this is the case if an acting entity, like a myosin moving or whatever, is constitutively relevant for a phenomenon like the muscle stretching, if and only if three conditions are satisfied. Namely, first, the acting entity is part of the system whose behavior or property is to be explained, which is our phenomenon. I explained that already. Secondly, the acting entity and the phenomenon are mutually manipulable. And thirdly, the changes induced by this mutual manipulation they are causally specific. What does that mean? So here we see a diagram that I think we saw at least in Alexander's presentation already. So the phenomenon, the muscle stretching, for example, the mechanism, the myosin's behavior, for example. So if we ask ourselves, okay, is this myosin's behavior, is this a part of the mechanism for the muscle stretching? What we need to do is first we need to find an operationalization of our phenomenon, which is usually in terms of an input-output relation. And then we here already assume the first condition is satisfied, so it is a part of the moving muscle. And then in order for the second condition to be satisfied, we need to run experiments. And the first manipulation is a top-down manipulation. That means we intervene into the input and see whether there is a way that by intervening into the input, we thereby change this component. So for example, I don't know, we make this muscle lift, the muscle has to lift, I don't know, a heavy weight, and then we see that probably what the actin and the myosin are doing somehow changes in some sense. So that's the top-down manipulation. In order to have mutual manipulability, we also have to perform a bottom-up intervention. That is, we intervene into, for example, what the myosin is doing, I don't know, block it somehow, and see whether this will change the output of the phenomenon. So in that sense, the muscle doesn't stretch anymore, or it takes more time, or whatever. And the third condition is it must be causally specific. So, for example, if you have a subject doing some muscle stretching and you remove the heart, the muscle stretching will probably stop because without a heart, a human can't lift anything. But this is not causally specific, it's just on-off. So our interventions have to be causally specific in the way that it changes the behavior, not completely, but in a way that is somehow, maybe proportional to how we intervene. So it must be more than just this on-off. What is important to the new mechanists, like Carl Craver, what they originally argued for is that this constitutive relevance already gives you explanatory relevance. So whatever is constitutively relevant is thereby explanatory relevant. Okay, so this is all I wanted to give you as a background on a mechanistic explanation. I hope it is clear enough, so what you have to, I will come back to that account and show you how it creates some troubles for other explanatory approaches. But now let's start with topological explanation and see how, what we can say about how this relates to mechanistic explanation. So what's the issue in general? So neuroscientists, and I mean, this is everywhere in science, they refer to topological properties in order to explain phenomena. Here's one example. So these are cases of a very abstract network with networks with increasing randomness, and it's often called, so this one on the left one, it has a regular topology. This one has a so-called small world topology, and this has a random topology. And especially this small world topology is supposed to be explanatory in many respects. So for example, it is said that the brain has a small world topology, and that explains why it's so efficient. Or even if the postal service has a small world topology, then it will be very efficient. Or a pandemic spreads so quickly because the main cities on the world are structured as in terms of a small world topology. And small world topology, just for those of you who don't know this concept, roughly means that things are connected in a way such that even though not everything is directly connected, you can still get from one to another relatively quickly by going through just a few connections. This is not supposed to be like the proper scientific definition, but this is what ChatGPT will tell you when you ask it what small world topology is. Okay, and now we have this like topological property. This is one example, small world, or having the property of being a small world network is a topological property. And now the core question is, well, do these explanations that refer to these properties, do they provide mechanistic explanations, or are they like their own type of explanation? And this is something we discussed. So Leon de Bruin is a philosopher from the Netherlands, and Linda Du, she's a system neuroscientist. And together we wrote this paper where we asked that question. And our case study was Alzheimer's disease. And this shouldn't be there. Yeah, this is very important, but I won't know. So this was a study on Alzheimer's disease. Actually, I don't remember what these symbols. It's in the DSM. Yeah, it's in the DSM something. Okay. So what these researchers did was to use multiplex modeling to find out how the topology of the brain is relevant to explaining Alzheimer's disease, or rather the cognitive deficits that you observe in Alzheimer's disease. And here, what they did, they used topological maps of the connections of specific neurons across different frequency bands. And here this is a toy description of the model they used. So this is something that we made up for the paper to get the idea across of what they did. So it's multiplex modeling because this model has different layers. And in each layer, every node in this network represents the same neuron, or rather the same voxels, but in different contexts. So these are the different frequency bands. So that you probably can't see it. So this is the beta layer, the upper alpha layer, lower alpha layer, theta layer, and delta layer. So different oscillations of these neurons. And now so they mapped, they took different subjects with Alzheimer's disease with cognitive deficits and patients with Alzheimer's disease without cognitive deficits, and mapped the same brain neurons or brain voxels. And then, in this model, they modeled their connections. And what you can see here is the dotted circles are the intra-layer hubs. So these are the neurons or the nodes that have the most connections to other neurons within one layer. And you have multi-layer hubs. That is, if you count how many connections, for example, this neuron number 2 has between all the layers, then you will come up with the multi-layer hubs. And this comes in degrees. And for example, here this is neuron number 5 has most connections if you could take all layers into account. And now, with help of this multiplex modeling, what they found out was that in Alzheimer's disease patients with cognitive deficits, the multi-layer hubs, so those with high centrality, so those that had the most connections within all or between all layers, those are damaged to a higher degree than they had in Alzheimer's disease patients without cognitive deficits. So these seem to be like the topological difference makers here, these multi-layer hubs. Okay, so this was our case study coming up with, here is a topological, or this explanation refers to a topological property of the brain to explain the difference between patients that have cognitive deficits and those that don't. And now, so what is a topological explanation? And this is a bit annoying, so the program seems to destroy my the font. But so here, Lauren Ross argues that so what is important here, I don't remember what was said here, but the second sentence is the most important. So that topological explanations, they capture a higher level structure that abstracts away from various lower level details. So it was just about the hubs, the details about the neurons and so on, or any other details about the brain didn't matter for that explanation. And secondly, the topology of the system is usually taken to capture a structure that is non-causal in the sense that it lacks the temporal information, so there was no time dependence here. And thirdly, according to Lauren Ross, most, not all, but most topological explanation, the dependency relation between the explanandum and the explanans is mathematical derivation. And now, yeah, luckily it shows the important bits. This doesn't sound very mechanistic. So mechanistic explanation, they focus on the lower level detail. They talk about the myosin and actin. It's causal, and the dependency relation is not mathematical derivation, but it's this part-whole relationship and the dependence. So this is why many argue, well, topological explanation is just some other different kind of explanation. It's not mechanistic. But as we argue in the paper, there are now different ways of how to integrate this. And as philosophers, we always in a paper have to first criticize someone else and show what someone else did wrong so that we can provide a better account. So here's Carlos Zetnick's account. And he is also a friend of the mechanistic account, but argues that topological explanations in some sense are mechanistic explanations. So he says that topological explanations are mechanism sketches that pick out organizational features of mechanisms. What are mechanism sketches? So mechanism sketches, they are explanatory, so they are still explanations, but they're incomplete descriptions of mechanisms, and they contain filler terms and black boxes. And the explanatory power of sketches increases if details are added. And at some point, you get from the sketch to the complete description of the mechanism by filling in all the details. And organizational features of mechanisms, so when we go back to this example, so the myosin and actin, they have to be organized in a certain way in order for the muscle to move. So the actin and myosin, they have to be somehow parallel to each other, and they have to be this temporal organization. So first there is the attaching to the other filament, and then there is the moving happening and so on. So this has to happen in the right temporal order in order for the mechanism to work. So organizational features here, this is the idea that mechanisms consist of parts that are spatially and temporally organized. Yeah, and as I said, the actin filaments are located in vicinity and parallel to the myosin filaments. The myosin filament first binds to the actin and then moves, and so on. Okay, so in that way, Sedgwick is arguing for the claim, well, topological explanation just is mechanistic explanation, but just very sketchy. But so I think there are at least two problems for Sedgwick's account. So first, topological features often are global organizational features. For example, small worldness concerns, so to speak, the whole mechanism, the whole network. So they are organizational properties of a mechanism as a whole. However, the organizational features referred to in mechanistic explanation are local organizational features, for example, being located near X. So they are features of individual components of mechanisms and not of the whole mechanism. The second problem is that topological explanations, at least scientists wouldn't think that they're incomplete in any way, that they need details to be filled in. Of course, there's more to be said about Alzheimer's disease and cognitive deficits, but as such, this topological explanation is, so to speak, it doesn't have a deficit, it doesn't need to be filled in. So that is, the explanatory power of this topological explanation as a topological explanation is not increased by adding more detail. So for example, the cognitive deficits in Alzheimer's disease arises because in Alzheimer's disease patients with cognitive deficits, multi-layer hubs are damaged. This is a fine explanation, and if you add details about, for example, specific connections and synapses between specific neurons, this won't improve this explanation, that it is irrelevant to this very explanandum. That doesn't mean that a complete model of deficits in Alzheimer's disease couldn't contain this, but this explanation is as such not, doesn't have to be filled in. It's already a good explanation. Okay, and now the account that we suggested is actually based on an account that Matt and I developed in our 2021 paper, and it comes with a few changes to the mechanistic approach. So we think that the mechanistic approach, as originally presented, cannot yet deal with this. But I mean, the new mechanistic approach is a very young approach comparable to how long philosophy exists, and also like people like Craver and Kaplan, I think with every publication make a few changes to the original account. And one of these changes Matt and I thought is a good idea, namely the idea that mechanistic explanation actually explain contrasts of the form the actual phenomenon versus an alternative phenomenon. And this is also, you can see in a paper by Craver and Kaplan. And Matt and I also introduced a distinction, namely between mechanism description, and mechanism explanatory text. And the explanation is in this third. And what we mean by this is, so mechanisms, they are the organized entities and activities that cause or constitute the behavior of the system. So these are the things in the world that you can manipulate and so on. The mechanistic description or mechanistic model or whatever you want to call it is the linguistic or graphic or whatever representation of these organized entities and activities that cause and constitute the behavior of a system of interest. And now this description as such is yet just in description. It is not yet explanatory. So explanation comes in at the level of mechanistic explanatory texts, and they can be said, yeah, they answer why questions that you can answer based on your description or based on the model you have. So these answers take the form of why is P actual the case rather than some P alternative. So for example, why does the muscle stretch at T rather than at a later time or whatever. And this means that we reject this assumption. So we say constitutive relevance is not explanatory relevance. Constitutive relevance gives you the description or the model. So by finding out which components are constitutively relevant, you can construct your model of the mechanism. But in order to answer these questions, you need some other notion of explanatory relevance. And this is something that Matt and I develop in this paper. It is a bit technical, but the core idea is that to provide an answer to questions of the form why P actual rather than P alternative, one has to provide a mechanistic explanatory text, and in order to get there, one needs to compare mechanisms. So just in brackets, this is like an ideal or a regulative ideal. So we're not saying that this is an accurate description of what scientists are actually doing, but this is at least what we think implicit in what they're doing. And so we argue that mechanistic explanations, so this mechanistic explanatory text, they mention only those mechanistic components, and now it becomes very technical. I will just give you a graphical illustration to make it 5 minutes. We started later, right? Okay, but okay, I would need 45 minutes or no, 45. Yeah. Okay, then I have to be really quick. Okay, so the idea is the following. So when we ask questions like why P actual rather than P alternative, we look at the actual mechanism that brings about our phenomenon, and it's now just a very toy example to get the idea. So let's assume that this mechanism consists of these components A, B, C, D, E, F. And now what we need to do is to look at all the alternative mechanisms. Let's assume this alternative phenomenon is brought about by, or could be brought about by these three mechanisms, and let's assume they are composed of these components. And now what Matt and I argue is that in order to answer your explanatory question, you have to find the alternative mechanism that is maximally similar to the actual mechanism, and this is in that case this, because it shares these three components and differs with regard to these 6 components, and all the other mechanisms are much more different. And then we have to detect all the differences that are shared by these mechanisms. So the difference to the actual mechanism that are shared by all these mechanisms. And in this case, this would be the lack of D, E, and F, and the presence of Y and Z. And this then would give us, so to speak, the difference makers between P actual and P alternative, and this then would be our explanation. So it is because D, E, and F is the case and not Y and Z, instead of not D, E, and F being the case, but Y and Z. And actually, it's much more complicated, but I will skip that. So the core idea is that these because explanations, they will become very complicated. You have to list all the differences that are similarities. So the core idea is that sometimes these differences can be summarized without loss in terms of topological concepts. So applied to our Alzheimer's disease case is, again, looks like I'm swearing or something. So why do patients with Alzheimer's disease show cognitive deficits rather than and others not? So that was the question that motivated this study, our case study. And in order to find this out, what they actually did is to look at this multiplayer network, multiplex model. So, and again, it's to get the idea. So let's assume this actual mechanism consists of A, B, and C, and at least one neuron N1 with N connections across 5 frequency bands. So now we can, for every neuron or every node, we can list how many connections it has at which frequency band. So it's purely descriptive. And now we compare that to the multiplexes that were derived from the Alzheimer's disease patients without cognitive deficits, and we can have a description of this, what is going on there. So they also have this neuron N1, but it has N plus I connections across 5 frequency bands, and we list that for all the other neurons, and we list that for all the patients. And now we find the one that is maximally similar, assume that is this one, assume that J is bigger than I and K is bigger than J. So then this one is the closest to the actual mechanism. And what they all have in common, these alternative mechanisms, is this plus, so to speak, so they have more connections, so just for this one neuron. So our answer to this question will be, well, they have cognitive deficits rather than not, because the N1, which has a high number of connections across the 5 frequency bands in healthy brains, has less connections across 5 frequency bands than N1 and NAD patients without cognitive deficits. We do the same for N2, N3, and all the 90, so they had 90 nodes in total. So we have this very long description of how the different, how the connections between these different nodes differed. And so the idea is, well, this is just a very long and complicated way of saying, well, multi-layer hubs have decreased centrality in Alzheimer's disease patients with cognitive deficits. So in a nutshell, topological explanations are mechanistic explanations, and so far as they pick out the organizational difference makers for a contrast to be explained P actual versus P alternative, where the difference makers are the similarities between the mechanisms that would have brought about P alternative, that are the differences between the mechanism that brings about P actual and the most similar mechanism that brings about P alternative, or in a smaller nutshell, topological explanations are mechanistic insofar as they pick out organizational similarities and differences between mechanisms that are explanatory. And given that I don't have much time, I just take 1 minute to show you how I think this can be applied to computational explanation. So I don't have to explain to you what computational explanation is, so I can skip that. And luckily, Matt and Holger presented the Lucas study. So as Matt said, this is one of the core case studies in our project, so I don't have to give you the details here either. Brilliant. And I can directly come to asking, well, is this a mechanistic explanation? And there's one problem, namely what we call the compatibility challenge, which is somehow at the core of our project. And this goes back to Mate's book, and he raises the compatibility challenge for representational explanations in his book. And the problem, I think, also arises for computational explanation. So for example, we could ask, is the property of the B and C region of these dendrites of the LGMD neuron, so they have the property of computing a sigmoid function. So we could ask, is this constitutively relevant? So this is, according to the mechanists, necessary to be part of the mechanism. And I won't go through all the 3 criteria because one, it suffices to show, no, this computational property or this description, this property cannot be constitutively relevant for the reason that they are not top-down manipulable. So there is no way to change the input to the LGMD neuron that would change what the B and C regions are computing. So there is just no way of changing the computation by changing the input. And now, my idea is to use this account that we used for topological explanation to make sense of how and when computational explanations are mechanistic. And I can only give you a toy application because so there is a lot to be said about the physiology or the mechanistic details about what the dendrites are doing. It has something to do with ion channels opening and closing and so on. I'm not at the point where I can give you all the details, but maybe just to get the idea. So the question here is, why did the locus jump at T rather than at T2, for example? That is a question that we could ask, and then we do what I explained. So we list all the physiological details about the dendrites, what they're doing when the locus jumps at T1. Then we list all the physiological details that the mechanism shows when the locus jumps at T2. And we do what I just suggested. We identify the maximally similar mechanism, then look at all the differences to this actual mechanism that are shared by all these mechanisms that bring about the jumping behavior at T2. And then we probably have a very long list of what the ion channels are doing and differences in ions moving and so on. And actually doing this will be a lot of work, but my guess is that, well, in short, what these mechanisms, these alternative mechanisms have in common that makes them different to this actual mechanism is, well, because the dendrites in region B and C did not compute a sigmoid function rather than computing a sigmoid function. But in order for that to actually show that this works, one has to correlate this computing of this function to physiological detail. Okay, there are many questions to be answered that will be part of this mechanism. For one, what is the mechanism here actually? And then with regard to explanation, is this just a pragmatic account, or is it really improving the explanation? Is this really still mechanistic? And does the case study really support this account? Is it really computational? That was connected to the problems that Matt talked about. And to what extent does the success of this idea really depend on which accounts of computation is presupposed? Because not according to all accounts, as Matt said, there's really computation going on in the LGMD neuron. Okay, and that's it. So the question is, can this be generalized? Thank you.
Thank you very much. It was really good.
Great, thank you for the talk. A lot of questions. Let me focus on the contrastive claim. Part of what you said was that when we are providing an explanation of phi, we're really providing an explanation of phi as opposed to psi by appeal to the actual mechanism as opposed to these other mechanisms. Is that claim, well, it seems to me when I'm a scientist in the lab, that's not what I'm doing. I'm trying to explain a focal phenomenon. I'm not considering other phenomena that are not occurring. And it seems like I can explain that phenomenon by citing facts about the actual mechanism that produces that phenomenon. It's a very general question. So were you proposing this sort of like, oh, this is what's actually going on underneath the hood? Or was this like a normative claim? This is what we should be doing? I was a little confused about the scope of that sort of description.
Yes, thank you. Yeah, that's a good question. Of course, often when you read papers, you don't see that they say, okay, and this is the contrast we are interested in. So, and I mean, this is a discussion on its own in philosophy of science, whether when asking questions, we at least implicitly always have these contrasts in mind. And one argument for that is that especially these why questions are very ambiguous. So why does the locus jump? What does it actually mean? Does it mean why does it jump rather than, I don't know, sit down? Or is it why does it jump rather than not jump? Or why does the locust jump rather than some other animal? So they are just, these why questions are very ambiguous. And so the claim is that, that at least implicitly, there's always this contrast in mind, which is usually often that obvious that it is not actually mentioned. So it's clear that if you do research on locusts, you're not interested in why does the locust jump, rather than, I don't know, the predator, which is nearby. So, but yeah, that would be the claim that it is something implicit to the explanantia of researchers.
Yeah, so that was great. So my question is, like, I guess a bit cheeky. Why is the topological explanation considered an explanation according to the criteria of mechanistic explanation that you described? So one of them, if I remember, was mutual manipulability, and that will require top-down and bottom-up intervention. But in the Alzheimer case, so perhaps there's like a top-down interventions, they need to do tests, but what is the bottom-up intervention and the graph theoretical measures that are being found in order to evaluate that this is basically satisfies the condition of mutual manipulability?
Now, good point. So I could have started this section with, in the same way as I started the computational section, and showing first topological explanations don't satisfy this. But here I started with Zatnick's account, so that would have been a further problem, that topological explanations don't satisfy this. So they are, so to speak, so the idea is that if you take constitutive relevance apart from explanatory relevance, you can accept, okay, it's not constitutively relevant, but still it's explanatory relevant because it has to be part, or it can be part of this explanatory text, and thereby you need this extra part of comparing mechanisms. So this is where topological and other properties come in. But yeah, so it's basically saying if you stick to the original account that this is explanatory relevance, then you can't integrate these different explanatory approaches because so many properties will not come out as explanatory relevant.
So in your account, you will deny the third mutual manipulability. So in your account, you will deny the mutual manipulability condition from the criteria of mechanistic explanation because you deny constitutive relevance as being related to explanatory relevance?
No, constitutive relevance still plays a role in finding these mechanism descriptions or your mechanism models, and then you compare them, and thereby you get all the other properties.
Any more questions?
Yes, it's not really a well-formulated question at this point, but I'm wondering what kind of criteria you use for determining which mechanisms are maximally similar to each other. So you had this example case there where it was kind of clear because we know the set of possible states that this mechanism can have, but if we talk about something like a neural population or even like a muscle, this state space is very, very wide. And I'm just thinking, from my perspective, it would make sense to reference topological models there because you can actually mathematically compare topologies and you do not need to know the detailed states of everything. You'd essentially be comparing models, right? Not like detailed to the last bit models, but is that something you're considering, or are you saying we have some other kind of approach there? Thank you.
Thank you. So I think there are two ways to ask the question that you just asked. So how is this practically done? And the other is more on a conceptual level, so how to, or an ontological level, so how to identify or individuate differences. So actually in the paper, Matt developed a nice algorithm to talk about these differences or detect this difference, or at least from on a conceptual level. Of course, on a practical level, that might not be, so as things are now, it's probably impossible because we usually don't have full descriptions of the mechanisms. In that sense, this is, as I said, so to speak, on the one hand, a regulative ideal, and in one sense, just it's an account of how to integrate these idealized pictures of mechanistic explanation and topological explanation. I think when you turn that into empirical practice, so observing what scientists are actually doing, so in this multiplex case, so this is in some sense what they were actually doing. So they were looking at the different multiplex models for these individuals and compared them and looked what are the differences between those models that describe the patients with the deficits, what actually distinguishes them from all the others. And probably all the other models from that modeled the topology of the neurons or of the brains of the patients without cognitive deficit, probably they were not all alike. Probably they also had differences. But these were differences that were not relevant to explaining this contrast. So in some sense, they were doing this already on this higher level, namely on the topological level. So this is saying you can compare these mechanisms already on an abstracted level. So they made these topological maps to compare mechanisms, so to speak. And I agree that this is probably what is often done because we don't have these full descriptions of the mechanisms. And this is just giving you an account of how to integrate them, like conceptually, these two accounts. This is not a description of what scientists actually do because they can't do that. We don't have full mechanism descriptions.
Great. So maybe we have one more quick question. Time for one more. Really quick.
Thank you. So basically, just to get it right, so you're saying that at least on the account that Oron advocates, this would be a problem for computational explanations, right? For probably similar reasons to the ones that Matt mentioned before. But do you think there's a more in principle issue here that prevents other computational explanations from being mechanistic?
So I think, thank you, that this problem is not yet depending on a semantic count of computation. I mean, with a semantic account of computation, you get into these problems that Matt mentioned. But so I think you don't even need a semantic account of computation when you're interested in is the fact that B and C are supposed to compute a sigmoid function, is this constitutively relevant, independently of whether you think there's representation involved or anything. It will come out that it is impossible to change that function by changing the input. So you can't, if you take like looming objects of a bigger size or whatever, this won't change what the dendrites are computing. It's impossible. I mean, that doesn't mean that it is that you can't change what the dendrites or the function that they are computing, but you can't do it by means of a top-down intervention. And this doesn't depend, I think, on whether you are a proponent of a semantic account or not.
Thank you very much. Yeah. So we will have like a 15 minutes break and then the next talk, right?
Yeah, then Markus.
Yeah.
All right, everyone. It is time for our last talk. And yeah, then I promise you 1 hour and then you'll be free. But
Let's see.
1, 2 hours, flexible.
It's open end now.
okay. So it is then my pleasure to introduce our last keynote speaker, Markus Werkle-Bergner, from the Max Planck Institute for Human Development, from the Center for Lifespan Psychology. And already in that affiliation, I think Markus's research interests are pretty much covered. Markus is interested in the development of memory throughout the whole lifespan and in neural correlates of developmentally constrained changes throughout the lifespan as well, using various methods, for example, EEG methods and others. So now it will be my great pleasure to hand the floor over to him. He'll be talking about brain maturation and memory development and approaches to study their interconnectedness.
Thank you very much, Matt, for this kind introduction, and thank you, Beate and Nia, for inviting me to present some of our research here. As you guessed from my affiliation, I'm not a philosopher, so I would rather consider this as a case study or several case studies that I present here. As said, I work at the Center for Lifetime Psychology, which is headed by Professor Ulmer Lindenberger, and there I direct a research group that is interested in how memory develops from childhood to old age. However, in the past years, for good reasons, we focused more on the child developmental side, and this is what I will talk about. So, what's the problem in our case? So by and large, what we are concerned with is a field that you can describe as developmental cognitive neuroscience. In cognitive neuroscience, we are typically trying to understand how the brain relates to cognition, or how cognition is brought about by the brain. And in the early golden days, in the early 90s, when MR scanners became available, many people thought, okay, now we can solve this riddle how the brain works, and we can find the brain area for emotions, we can find a brain area for decision making, we can find a brain area for love, and we tried to find an easy mapping between cognitive states, like we describe them in our daily lives, and brain regions. And as most of you might know, nice attempt, but it mainly failed because this is typically not how the brain works. So maybe what we need is a kind of a common model that describes how cognition and the brain work interactively. But as it turns out, this is a very huge endeavor, this is a huge challenge, and we are far from reaching it. So maybe we should be a bit more humble and try for an intermediate ground. So probably what we can currently do is that we can find a mapping between cognitive processes and neural mechanisms, or neural processes, how you want to describe it, which might or might not relate to computations and or biological mechanisms that we find on one on the other side. And this is where we also meet in the representational ground, because this is finding mappings between cognitive process representations and neural mechanistic representations. And maybe on the long run, this can also be integrated in a common model, but currently we would be okay if we find good enough models on this middle ground. So, but this is more or less the approach of cognitive neuroscience. We can add another level on it if we go the developmental route. And my research team and we started out with a more or less simple question. We asked, okay, how does memory develop, and how might this memory development during childhood depend on, in this case, the entorhinal hippocampal system? Very easy question, very straightforward. The task is fairly difficult. Yeah, but it's already difficult enough to find a correspondence between cognitive processes and neural mechanisms in a static system. Here we deal with change, with development across time. This happens not just in the behavior, the targets that we want to study is changing constantly, but also this target is influenced by what happens on the environmental level, on the societal level, but also on the brain level. And the brain itself is changing constantly. The brain of a 1-year-old is not the same brain as the brain of a 90-year-old. Also, it might be developmentally related if it's from the same person at one point. Yeah. So here we need to find a mapping between change dynamics in the best case scenario. What are the prerequisites to get at these change dynamics? Typically what we need to do is we need to observe change. There is no shortcut around it. For a long time, and still today, the prevailing paradigm is cross-sectional studies. Yeah? You invite people from, let's say, 1 to 100 years of age, and you give them a bunch of tasks, and you check how different they are, and you might think, okay, now I found something, how this skill, this task performance might change from childhood to old age. But this is typically not the case. What we have here is simulated data. So here are 75 individuals, simulated individuals, where we know by simulation that this is a linear change with some random Gaussian noise around it. What you now do is, when you do a cross-sectional sampling, is you send them randomly from these change trajectories. Yeah? And if you take it away now, what you get is a bunch of data, and depending on your sampling process behind it, any type of change trajectory can be estimated into such a cloud, depending on the cloud, how the cloud looks like. Yeah? So just from eyeballing here, if I do a survey on it, some people say, okay, this is a quadratic change. Some people say it's no change at all. It might be a linear change. If you are unlucky with sampling in the upper part, you can also get an increase over time in such data sets. So by and large, these cross-sectional data sets might be a good hint towards what we're actually observing, but they cannot tell us about how change dynamics relate to each other. Yeah? So if you want to observe change, if you want to know about change dynamics, we need to observe change. And in humans, this takes time. Also in animals, but in humans especially. So by and large, what we need for a more or less successful approach to developmental cognitive neuroscience is, on the first hand, good models. To be honest, in cognitive neuroscience, good models are disputable. We are not yet at a level of understanding approach avoidance in locusts, for example. This is not yet what we can do in cognitive neuroscience. Typically, we have models that are good enough to describe the phenomenon. This gets even more complicated if we talk about change or development, because there is not much data around that shows us how things change in a given individual. We still need to create these data sets. This is the second thing we need. We need good data, and good data means good tasks and good descriptions of the constructs we want to study. What is typically done is that you develop a specific task, like a word list recall task, and this recall task stands for memory. But this might only be one facet of memory. Memory might be more complex. So probably a better approach would be to use multiple tasks and multiple indicators to get at a certain construct of interest. And finally, what we need is observations across time. And here time depends on what you want to observe. If I want to see change from birth to 2 years, I have a 2-year observation period. Yeah? This is fairly small. But if I want to study change in adults, we talk about observation periods from 10 to 30 to 50 years. Yeah? This is what it's needed to cover the range of what we are typically interested in. And we are all scientists, we know how funding works. With the DFG, for example, you get a 3-year grant. Yeah. If you are lucky, you can do a Sonderforschungsbereich, and then you might have 5 + 5 years or something in this range. Yeah. 50 years, 10 years, difficult. Yeah. And in brackets, this is why I shifted to children now, because the observation periods are smaller. And my career is going to an end, so I should speed up a bit. Okay, so let's start with our first research example. I will now introduce 2 studies we are currently running and will show you how we approach these problems in understanding how memory might change. And the first one is studies that ask for the development of memory specificity and generalization in childhood. Here we start with the observation that children are extremely good learners. Even in infants can, from some examples, acquire a whole language over time. Yeah? Older children learn grammar, learn spelling, learn writing, and all this stuff, despite the fact that typically, especially infants, are very poor in remembering when, how, and from whom they learn something. Yeah? Memory for details of a given episode develops slowly. Only in the late childhood, early adolescence, we get a full grasp of our experienced past. While if you ask kindergarten children for what they had for lunch, you typically get something like noodles. And even the question which kind of noodles with which kind of sauce is difficult. Yeah? So there's a huge gap between what children can indeed achieve and how much they remember in details from it. How can we, but nevertheless, we believe that this tension between remembering details and being able to generalize from based on memory is important. An adaptive memory system needs to find a balance between these 2 affordances. If I meet a friend and he suggests, okay, let's go to the restaurant we met last time, I should be able to remember that it was the Italian place and not the Chinese place where we met last time. Otherwise, I may be lost. I need to dissociate 2 comparable memories from one each other by means of distinguishing the details of it. Also, if he asked me, do you remember last time we talked about X and Y, I should be able from this partial information to recover the entire discussion we had to fill in the details that he might refer to later. At the same time, if I know that I meet this friend at an Italian place, at another Italian place, at a noodle place, at a pasta place, and a pizza place, I should also be able to infer on the long term that whenever I invite him, I should suggest an Italian place because he seems to prefer Italian over Chinese restaurants. Yeah? These are different affordances, and one time I need to extrapolate from several experiences and disregard the details, and another time I need to remember exactly these details that I typically ignore. Yeah? And a good adaptive memory system needs to find a balance in between. And we ask, how does a good memory system develop this balance from childhood to old age? To get started, we draw on computational models developed in the field of cognitive neuroscience, especially the complementary learning systems framework, which would suggest that part of this task is solved by the hippocampus by providing different computations. On the one hand, within the hippocampus and within the substructures of the hippocampus, especially then the gyrus is well suited to perform something like pattern separation, to identify that 2 overlapping information patterns are indeed to be separated. They can be orthogonalized via representations in the dentate gyrus. On the other hand, the conoharmonious fields in the hippocampus are well suited to perform another function which is critical to perform pattern completion, to derive full experiences from partial input. Yeah? These are things that are typically on a one-shot, fast processing lane, whereas generalization across several experiences requires some longer timescales. Yeah? Here we have the interplay between the neocortex and the hippocampus that might provide such a tool that in the neocortex, longer-lasting representations can be formed that are abstracted from the details of a given episode and perform the basis for generalization. How can we get a glimpse on this? One concrete example of our research, we used the so-called mnemonic similarity task to assess the amount of pattern separation in children, adolescents, and young adults. The image is a bit blurry, so this will get a bit more difficult. What typically is done in this task or in variants of it is that our participants see a series of objects. They try to remember these objects, sometimes with a shadowing task here, whether it fits in a box, yes or no. And during retrieval of these items, they are presented with different kinds of these objects again. Sometimes it's an exact repeat of the object, yeah, for which they should say, okay, this is the same thing that I have seen during learning. Or it's a similar object, here like the duck. Yeah. This is the original duck, this is the duck during retrieval, this is a similar duck. Yeah. And there are some new objects they did not have seen before, and they should say new to these things. And what we can do is we can compute a measure which gives us an idea how much you engage in pattern separation, dissociating similar objects as actually separated from each other, or how much you engage in pattern completion, trying to conclude the same object from a similar input. If we provide this task to children, 6 to 10 years, adolescents 11 to 14 years, and adults, what we see is that this ability to, or this tendency to engage in pattern separation processing increases with age. Yeah? This is not a surprise, this is known for quite some time. But critically, at this point, we also scan the brains of all our participants, and we use some, at this time, modern sequences to get at high resolution images of the hippocampus, which is shown, let's do it here. This is the hippocampal region, zoomed in from here. And based on tedious manual tracing, it was possible to separate out the different subfields of the hippocampus, at least in parts, to get such 3D volumetric estimates of hippocampus for the different participants in this age. Under the assumption that the hippocampus does not, or the subfields of the hippocampus does not increase or decrease independently, but that the hippocampus is situated in confined space, we applied a multivariate technique, so-called partial least squares regression, to understand how much change in the, or how much differences in the volumetric measures of the hippocampal subfields can be attributed to age. So how much age variance can be predicted by knowing the structure of the hippocampus. And what we found out is that we get indeed one reliable profile that suggests that with increasing age, you have a larger structure, a larger volume of the dentate gyrus CA3 region and a reduced volume of the entorhinal cortex region. And the more our participants express this profile, the more they engage in pattern separation, in line with the idea that the dentate gyrus indeed is one of the key regions to perform pattern separation functions in memory, and that the change or the differences, the age differences in this region are related to the ability to perform this function. Yeah? But, full disclosure, this is cross-sectional data. This is just a one-shot observation which gives us an idea about how age differences might play out and how neural differences might play out here. Based on behavioral observations that I just described and based on those and similar findings with colleagues from Temple University, we came up with a hypothesis that states that the hippocampal maturation drives our memory from initially preferring generalization towards specificity with increasing age during adulthood. Yeah? So we assume that early in development, it's more important for children to be able to generalize at the expense of extracting specificities. So if you are born, probably it's good first to learn the rules before you start to differentiate the rules from each other. And we assume that this really depends on the maturation of intrahippocampal structures. But as you might directly recognize here, we have cross-sectional data and we have a change prediction below on the theoretical level. Yeah? There is a clear mismatch. So for the past few years, I think we started in 2018, we formed a research program that tries to test these predictions in a more formal way. These are our so-called comic studies, which come in two flavors. On the one hand, a longitudinal study, and on the other hand, a psychrometric study. So I told you what we need is good models. So here we have a fairly good model, computational model for the phenomenon we are interested in. We need good data. This is why we run this psychrometric study to get a good idea of the psychrometric and the measurement properties of the tasks we are using. And we need observations across time. This is why we also run a longitudinal study. Yeah? If you're interested in the measurement study, let me know. In the end, we can talk about it. For time reasons, I will focus on the longitudinal part. What do? We implemented a 3-year longitudinal study with an accelerated longitudinal design. So we sample kids from 4 to 8 years at our first measurement time point. We aimed for 200. Full disclosure, we didn't get all 200, but we get close to it. And we repeat the measurements in a 1-year gap for 3 years, thereby covering the entire age range with the possibility to test for retest effects, because we have kids who do the task only once in each age group, and we have kids that do it repeatedly, and we can separate repetition effects from change effects here. What we do at each measurement time point is that our children come for at least 5 sessions, so 5 different days. And we run a bunch of memory tasks, of questionnaire tasks, and intelligence indicator tasks. This is one part of it. Critically, we also do measurements of sleep EEG and actigraphy across at least an entire week to get an idea of the habitual sleep patterns. Here we are in the environment of what is happening. And crucially, we do MR testing. So the kids come to our lab. One session, they get acquainted to the MR facility. For this reason, we have a so-called MOG scanner. This is a MR scanner that is exactly an MR scanner, but without the strong magnet. This is an easier environment because parents can get in. There is no risk due to the magnetic field, and we can help the children to get familiar with the entire environment. And last day, they go in the scanner, and we run several structural MR sequences to get at full brain structure, high resolution structure of the hippocampus, and if possible, to connectivity. But this is a sequential process. The connectivity, the diffusion-weighted sequences are very loud, very long, even for adults, sometimes frightening. So not each child is doing it, and to keep them as motivated for as long as we can, we start with the easy sequences and do it in the end. So we know that we have a lot of missing data here. And this is what you have to live with. We lose a lot of data due to children not being able to complete all the measurement steps. Yeah. What do the children do? The main thing they are working on is our so-called collection game. When we work with kids, we always try to come up with an overall narrative that keeps the children interested and motivated, ideally in a kind of a gamified version, not in a computer game, but in a real game with boards and so on. And here, this is the collection game, and we introduce it in a way that they see a bunch of images where they always consist of an actor, environment and an object. And what the children know is that each of the actors likes to collect things that fit their collection. Yeah? And they should just watch what they get presented. But hidden in the structure, obviously we are psychologists, so we do hidden things in what we present to them. There is a structure to it in a way that each actor likes to collect items from a certain thematic region. So for example, the actor here in the top row likes to collect musical instruments. This actor is interested in kitchen stuff, and so on and so forth. What we can do now is that from this setup we can derive a set of measures. On the one hand, we can ask our children for generalization abilities. So we present the actor and some objects that the kid has not seen so far, and the question is, okay, what would this actor like to collect? Anyone? The piano. Yeah, in this case. So this would give us an indication that they can indeed generalize the overall structure. We can ask for the exact pairing, which is called, in our case, context binding. So this actor on this scene has seen which of these objects. It's this one in this case, so we get at precise memory. And we can ask for specificity on a conceptual and perceptual level to derive also this kind of measure. So we can ask what kind of instrument this kid has seen here and what the color was, for example, yeah, to get different aspects of specificity. We ran this task initially. This was an adaptation from a task we got from the Temple University from the US. So we translated everything to Germany. So we had to do a validation study, which took place during Corona, so half in lab, half online. And the good thing is that we can get behavioral evidence that is in line with our assumptions that age differences are more pronounced and stronger and faster for the generalization material instead for binding or specificity measures. We did this with a pilot sample, and some months ago we finished our first measurement time point. So we are lucky here, and this is now a pattern separation task also for us, because this is the original data that I have just shown you, which is published from the validation study, and this is the cross-sectional data from our first measurement time point, and it's basically identical, which is relieving for us. After more than 1.5 years of data collection, we at least know that we did not mess it up. So here we are quite happy. What's the goal of this entire exercise in the end? We are currently in the middle of our second measurement time point, and some weeks ago, the first kid of our last measurement time point started. Yeah. So now, after we started in 2018, some years, we now finally see some light at the end of the tunnel. There is indeed an end of the study, and we aim for summer 2026 to complete data collection. So we are still in the middle of it, just to give you an overall view how long this takes. In the end, what we aim for and why we started the whole thing is that we want to understand the lead-lag dynamics between changes in the hippocampal fine structure and the development of memory component processes. For this task, what we need is crucially these 3 measurement time points, because they allow us to fit models like this one, which is a conceptual model in this case, an instructional model, a so-called latent change score model, which allows to model not only how the measurement at one time point, let's say on the neural level, is influenced by its previous time point, but also how the measurement of cognitive processes might influence the change on neural processes or the other way around. These latent change score models in a multi-time point context allow for the assessment of lead-lag relationships. This is something that you do not get with only 2 measurement time points, and this is something that you do not get with only 1 measurement time point. You cannot assess how one change in one variable might predict and precede the change in the other variable. This is why you need multiple of these time points. Yeah? This is what we aim for in the end, but given that we need some more months to finish the study, I hope I get invited back in 5 years from now and I can tell you the final story about it. Yeah? Take a note. So this is one example of it, and one example how complicated this approach and how much time it takes to actually implement it. Another example, which is slightly different, starts at the beginning of our lives. Here we combine human and mouse studies to understand memory engrams, and we make use of a specific phenomenon as a model system here. What is a memory engram? This is an idea introduced by Richard Salmon already at the beginning of the 20th century. Salmon is or was a German evolutionary biologist, and he was concerned with the idea how ideas and memories can be transmitted across generations or even across time. Yeah? He was inspired by Darwinian thinking and the idea of genes and everything, and how this is shaped by evolution. And he thought about, okay, how could this be done for our experiences? And he came up with the idea of the engram, suggesting that, okay, what is needed to transmit something across time, our experience across time, is that they lead to an enduring change in the physical substrate. Nowadays, we would say in the neural substrate. So this is experience specific, and that it can lie dormant for quite some time. Yeah, we do not remember all our experience all the time, but we can retrieve them when we need them. Yeah, and this is the last critical thing. It should be the basis for memory recall. Interestingly, this happened more than 100 years ago at a time when it was impossible to test for it. Nowadays, there is an increase in interest in this in engrams, because now we have the molecular techniques to tag individual neurons that are involved in forming a memory. And with optogenetic techniques, we can also manipulate them in animals. Yeah. Typically in rodents, but also in other animals. So we teamed up with one of the experts in the field, Thomas Ryan. One of his PhD students, Sarah Power, switched labs and switched fields from animal research to human research, and we created our so-called ECHO studies, the early childhood memory studies, which are intended to understand how memory engrams might be formed in humans, and we do this by trying to understand the phenomenon of infantile amnesia. Infantile amnesia is a naturally occurring forgetting state that affects nearly everybody, which just describes the fact that most of us cannot remember things that happened in the first 2 to 3 years of our lives. Yeah? Crucially, this is not just in humans, but in several animal species. This is also the case, amongst them rodents. Also mice show infantile amnesia. Yeah? So we thought, okay, this is a good starting point to do a cross-species study and to use the advantages of engram labeling technology in mice and try to translate it to humans and the other way around. How does this work? So the first crucial thing here is what Thomas Ryan and his lab and also other labs nowadays could demonstrate is that you can label memory engrams in infant mice. So infant mice undergo, in this case, a fear learning procedure, and they create a memory for this fear event. And typically, if you keep the mice for long enough, they start to forget about this event, and they do not show any freezing behavior anymore, which is the indication of infantile amnesia in this case. However, if you use optogenetic techniques, when these same mice get at the adult stage, non-treated mice do not show this effect, but when you optogenetically stimulate the engram, the fear response comes back. So this would suggest that, contrary to common belief, infants can lay down memory traces, and these memory traces are not what the term amnesia would suggest. They are not lost, but they still persist in the system. They just cannot be accessed anymore at adult stage. But if you find the proper way, it's possible. Yeah? So they might be still active. So this is a good paradigm. The first objection is how to translate it to humans. Would you bring your 1-year-old kid to our lab for a shock fear procedure? Okay, we have the equipment, but we don't do it, so we need to find an alternative approach. Luckily, Thomas and Sarah found a different approach. They used the so-called Barnes maze. The Barnes maze, here shown for mice, is an open field maze where there is one hole in the surroundings of the maze, and this is an open field, and so mice typically, again it's fear, do not like to be in an open field, so they try to hide. Yeah? But given that there's only one spot to hide, the mouse has to learn over repeated trials where this spot is located. You can combine it with contextual clues, so that given a certain environment, the mouse learns that in this environment the spot is here, in this environment the spot is here. Yeah? So we can do location context memories, associative memories. What Sarah has shown in her dissertation work is that on the one hand, yes, mice show forgetting, they show infantile amnesia for this type of learning. They can learn it, they can remember it on the immediately following day after learning, but they forget it after 8 days. And it can be optionally reactivated. So here also there is a trace for it that persists in the system, which is not naturally accessible, but can be optionally reactivated. Yeah? So here we have a model system that is also manageable in humans in this case. So what we did is we created a human Barnes maze at our institute. This is schematically shown here, and with one of our pictures. What we have is a 4x4 meter room, which has walls that serve as screens for projection. Yeah? By this, we can switch the environment on a trial-by-trial basis. And we created very unique environments, very unique stimuli that only exist in our lab. So we are more than confident that these kids do not experience any of these things somewhere else. Yeah? What we do then is we have to adapt it slightly. So we do not work with fear in this case. We have such toy puppets. They are of this size. They are also handmade for our purposes. Sarah draws them, sends them to a company, and the company builds them for us. They can also make sounds to a certain degree, and we have these small boxes here, and we introduce it to our participants in a way that we say, okay, this is Corky, and Corky likes to play hide and seek. Now let's learn how you can find Corky. And given that this is an interesting toy character and makes sounds, the children like Corky and they like to play with Corky. So over repeated trials, the children learn that in one environment, Corky likes to hide in this box, and in another environment, Corky likes to hide in this box. Yeah? We fully adapted the mouse design in having one first session where they just do the learning. They come back 24 hours later, where we confirm that the memory still persists. And then was the question, okay, how long do we need to wait until children forget? And we asked several of the most prominent experts, and the answer was between 1 week and 12 months. So where to start? We did not know, so we made it part of our design. So we have our kids coming back either at 1, 3, or 6 months at the moment. Now we are contemplating about 12 months, having as an additional condition for good reasons you will see later. But this is how it currently works. And the sessions on the second and third session are exactly the same as the first session, except of the fact that we stopped the children from exploring the environment after they opened the first time one of the boxes. Yeah? What we can get from the entire thing is that we can get information on different kinds of memory. We ask them for the memory of the object, of Corky. Yes? we have several of these toys. They are most of them are living in Sarah's office, and we can ask the children, okay, now do you remember with whom you played yesterday? And they can point to it. We also have several of the scenes, and we ask them for, okay, do you remember which scenes or which context you have played in yesterday? So we get at object memory, we get at context memory, and we get at object, context, location memory by having them playing the game again. Yeah? So if they learned that Corky likes to hide in this spot, in this environment, after we bring them back in the second and third session, they should ideally immediately go to this box to find Corky. Yeah, yeah, this is a huge endeavor. It also takes long. If we wait for 6 months until a kid can come back, this adds up. This is also running for 3 years now. The lab is quite a fun lab, so recently also Science was interested in the topic and also highlighted our lab in one of their journalist contributions here. But the critical part for us is now, after we have done all this exercise, do the kids really remember something? Do they really form a memory trace? And this is hard to tell because different to mice, we do not play with fear in this case. So we play with motivated behavior. And whoever has worked with 2-year-old kids, motivation is a random target. Yeah? So there are several reasons why a given kid might go to the right box, yes or no. Yeah? So we have a huge variety and a huge individual differences among the kids. So we need to convince ourselves that we indeed were able to install at least some kind of memory in our children. And the first measure we came up with is the idea that, okay, independent of the context, there are 2 critical boxes. One, which is the correct one in the given context, and one which is correct but in a different context. But there are always 2 boxes that are always wrong, independent in which context you are. Yeah? If I have no memory at all, I should select all these boxes randomly and more or less equally. Yeah? This would suggest that we have a 50% chance to commit a random error, yeah? If we do just random behavior. If you at least remember the wrong box in this context, but which is right in the other context, we could argue, okay, you might have some kind of memory, maybe not the correct one, or maybe you were led wrong in this case, but there is some kind of memory. So what we look for now is to say, okay, how many trials do you commit a random error? So how much time are you really wrong? And if this is more than 50%, we would argue, okay, this is random behavior. Yeah? These kids do not remember anything of our task. But luckily, it seems like this is not the majority of children. Rather, it looks like that the vast amount of kids is quite good at remembering at least parts of what happened. So this is already some evidence that we were successful in implanting memories in our children. Similarly, we can look at the learning behavior. Yeah? If we plot, again, this random contextual or no error measure, or the number of boxes they opened, we would assume that over repeated encounters during learning, the number of correct choices increases, which is seen here, while the number of random opened boxes decreases. Yeah? From this data, for the middle age group and the oldest age group, we would infer, yes, there is some kind of learning going on. So we are quite confident now that at least in some kids we were successful in laying down a substantial memory trace, a substantial memory engram. Admittedly, there are huge individual differences, and we need to understand why some kids do learn and others don't, and why some kids learn the full range and others only part of it. But we can also look at the forgetting. This is what we were initially interested in. And what you can see here is, again, this is partial sample because we don't have the full data set yet. We are in the midst of data collection. We hope to finish this study by the end of the year.
But what is already getting more and more reliable is the observation that obviously the age at encoding and the delay decides about how long you maintain information of it. Our youngest age group seems to forget quite fast. Already at 3 months delay, there is no deviation from chance. This might be also the case for 20 to 22 months olds. Interestingly, our oldest age group also forgets about after 30 days. Yeah? This is unexpected in our case, but at least there is an emerging pattern that we still need to understand fully, that the age when you encode a specific object location memory and delay when you are tasked for it play a role how much you can naturally retrieve from this experience. I hope that we can get closer here. And how do we? How could we reach more information? How could we elucidate the individual differences? Yeah, the lab has more features to it. Despite having such a fancy environment, we also have cameras on the ceiling. The cameras allow us to track the full motion of our children while they play these games with us. And we make use of recent AI-powered technology in markerless pose estimation. So we use deep lab cut to trace our participants while they perform the task. And this here is one example how this looks like. You can see this is one of our children, and this is obviously a quite well-trained one. It immediately runs to one box where it assumes that Corky is hiding in this environment. The environment we cannot see, but we know from the log files which environment this was. And as you can see, we can trace the entire movement trajectory of this kid. And Deep Lab Cut is smart enough to also separate the children from any other person in the environment. Yeah? So we can get rid of our parents and our experimenters. We just get the behavior of the children in this case. And what we now can do is we use analysis strategies that tell us something about the search strategies that might have been employed, so based on the search trajectories. If I have no clue at all, I may randomly open any of the boxes. Yeah? If I know that Corky is hiding in this box, in this environment, I may go straight. So these are the two extremes. There are also some procedural strategies. We also do the same thing with adults, with more boxes in a slightly different environment. Some adults just go box by box in the first round. Yeah, this is a procedural strategy to solve the task. And we feed the movement trajectories to the RTrack software, which analyzes the movement trajectories, and we can separate out different search strategies. And also here you can see that over time, so over repeated trials, in the two analyzed age groups here, the number of more or less random search strategies here in the orange-yellow colors decreases, while the more directed search strategies take over. Now we can start to create models that tell us how much of the search strategy predicts us about the later success in retrieving our memories. What we try to achieve with this type of strategy is that we get a more or less comprehensive phenotyping of learning and memory in these 18 to 24 months old children. We have our dense behavioral data from 2 sessions, and we have our, oops, this is going too fast. How do I get back? This way. We have our video data. We have some background information on the children, so we have some parental questionnaires, we know about their language status, we have some questions about their motor development to get at the background of the children in this case, and crucially we also run mobile EEG recordings on these kids. So at least a subset of the children is equipped with a 32-channel EEG. ideally in all 3 sessions, and we would like to use the EEG decode the specific object location memories acquired during these learning times. And in the long run, we would like to translate this back to our mice, where we also would like to get at EEG signals of specific engrams and to compare ways to identify neural markers of our engrams in children and mice. So in the long run, the goals of this research is to identify the sources of individual differences in the expression and in the duration of infantile amnesia as a model system for naturally occurring forgetting that is easily accessible in humans and in animals to allow for cross-species comparisons. In the very long run, we would also like to develop non-invasive techniques for engram identification and ideally also for reactivation in humans. Yeah. There are some ideas that need to be validated first in the animal system, but if we have this translational platform, we would have a means to go back to the humans and maybe try some really safe non-invasive techniques to reactivate specific engrams. Last part is a side note here. This might all be linked to immunological challenges during pregnancy. This is what we study together with Claudia Buss. This is also ongoing work here. So by and large, these are 2 examples of how we think we can approach this question of how memory changes during childhood. These are 2 different research strategies we employ, one really with longitudinal assessment, the other with cross-species approaches. And so at the end, I want to thank my team. As you can imagine, I'm not the one who's doing all the tedious work in the lab. It's these people here, the postdocs and the PhD students. Anka Jöchner and Lisa Buchberger sadly left the group some time ago, but some of the rest are still working with us, and they are really pushing hard to get all this data in and to finish the studies in time. And I also want to thank our dedicated student assistants. We have a fairly large team because to take care of all the kids, you need at least 2 persons always be in room, sometimes 3 or 4, depending on what we currently do. So we need a fairly large team here, and I'm very happy and thankful that I have the pleasure to work with such a dedicated and inspiring team. And I want to thank you for your attention.
Yes, so thanks, Markus. And now we have about 10-ish minutes for questions. So, Jonathan, also. And then...
Hi, that was fascinating. I have a lot of questions, but I will only ask one, which is a methodological question. So, like for identification of engram, you usually need to label the cell population, usually using opsins or some other way. How can this be done in any way in fMRI, EEG methods, or is that basically non sequitur? There's no way to basically identify the initial cell and how, or initial cell population, how it changes. And do you think it kind of raises some tensions in identifying engrams in humans because of that?
Yes, this is one of the huge challenges. Obviously, we cannot go down to the single cell level, and for good reasons, we cannot do genetic engineering or molecular tagging. This is out of reach for good reasons. But there are more indirect ways here. There is some kind of localization of the engrams, but many tagging approaches currently would suggest that the engram is more or less a whole brain phenomenon. There are hubs, because we know that you do not need to stimulate the entire engram, but it's sufficient to stimulate in certain hub regions, maybe hub regions. But it seems to be, at least parts of it seem to be widespread. We don't know yet how many, how much, but this would open the gate for some more mesoscale approaches like fMRI, EEG, MEG. And over years, yes, with multivariate analysis techniques, there are already some ways how to get at some kind of engram here by showing similarity of activation patterns in fMRI during encoding and retrieval between different stimuli and so on and so forth. Svenja Brod and colleagues showed via structural MRI, via diffusion-weighted MRI in gray matter regions, that there might also be a structural correlate that might change in the parietal cortex. These are more or less proof of principle studies that would suggest, okay, there are ways how to get at it. Here we would have a very unique opportunity if we have indeed two parallel lines of experimentation in animals and in humans. We could try to use the multivariate techniques in our humans and go back to the animals and do optogenetic stimulation while we record EEG and try to get at similar measures. And in an ideal, theoretically ideal world, we might find even a common space where these engrams live. This is today science fiction, but at least there are ways how you can imagine how this could work. Whether this is possible given real recordings, real signal-to-noise considerations, and so on and so forth, I don't know. But at least we have a means to try it now. Yeah.
Next was Fatma.
Yeah, great talk, thank you. I guess my question goes towards change dynamics question. So when you do these longitudinal studies, right, there is several aspects to the change. One is the brain changes, just because we are plastic, right? Then it changes because you have a task that your kids are learning related to plasticity, but unrelated to the task, there's already plasticity. And then there's also the effect, like the aspect of like having changes because of you're taking the measurements from your scanner in different time points, which is measurement noise. So my question goes towards like, how do you control towards these things, which is, I know, a very difficult task. I'm thinking about the same question. So I wonder if you have answers to these three.
These are indeed the crucial questions that everybody working in this field has to deal with, and there are only partial answers to it. We try to control what we can control. Yeah, when it comes to the measurement noise, so the MR scanner, we have the luxury that we have a dedicated research scanner which undergoes constant quality control. So we at least know when there are some major changes in the field, in the thermal noise properties of the scanner, and so on and so forth, which we could integrate in our models. This is, I guess, as far and as best as you can do it. Otherwise, it's something you have to live with, that there is some kind of noise. It goes even further when we think about how to do the segmentation of the hippocampus. Do we use manual or automatic segmentation? If we go for manual segmentation, or at least partial manual segmentation, do we use the same raters across time, which would mean to hire people for 4 years? Do we use some replications of it, and so on and so forth? This is all a continuous problem in doing this type of research. What we hardly can control is any plasticity in the brain beyond what we measure. So some kids do sport, others don't, and so on and so forth. There is also some plasticity noise, if you want, to it. What we can control in part, what we also do due to experience with other longitudinal studies, mainly in adults, is to try to control for retest effects. This is indeed a huge problem, especially in memory tasks, longitudinal memory tasks. They can go in both ways. They can increase or decrease performance over time. Maybe due to increasing the types of errors, the intrusion errors that occur. This is what we try to control in this accelerated longitudinal design. So here we can check whether the performance of, let's do it this way, show it from one example. For example, whether the kids that are 8-year-olds and who do the task repeatedly, whether this is comparable or is ideally the same as the kids who do it the first time. Yeah, this is how we can, in part, control for the retest effects here. But yes, this is a huge problem, especially in memory studies of this longitudinal type.
A follow-up comment maybe on the measurement noise, because I mean this also happens without the longitudinal actually, right? If you do just have one task session, one experiment with several sessions, that can also happen. And I think one way you can control for it is, in fact, to include repetition of the same task, or independent of your task even. You could give them some story, let them read it, or like listen to it, 5-minute snippets in that session, and the other session, and another session, and then you can look at the signal-to-noise ratio across these different sessions to see, is there a real change? Because even if there's quality control, the brain just, like this, there is just measurement noise that comes in, and then you can have a ceiling for your SNR basically.
Yes, this is what we do in our measurement study, in the accompanying psychometric studies. There we had to have a larger battery of tasks, and we can check for differential reliability of our tasks with regard to the measures of interest. This is a separate 200 children psychometric study that we run on the site. Here we cannot do it due to the time constraints. The task, like it's set up currently, already takes 40-something minutes, which is at the top of what we can do with 4-year-olds in this case. So we can hardly repeat it across time.
And finally, Holger.
We especially do the main tasks always in the middle of our procedure. So they come for 5 sessions, and the main task happens in between, where we have at least some adaptation to this whole environment to being tested, that we get rid of all this unspecific noise which happens due to the fact that you come to a lab. This we try to reduce by having the critical things in the middle.
Thanks very much. Excellent. One question you all may almost addressed already, that's the retesting effect. I mean, in theory, you could have 2 groups if you had, enough experimental participants. Then you had one where you have a different interval of retesting, and then see whether that over time makes a difference in the outcome. Did you have in your auxiliary slides the one where they tested grid cells using fMRI and Newman? So that would be, I would imagine, you might as well be able to get the n-gram in children if you would test them by fMRI.
I got you right. You test to test for grid cells.
I saw in your when you were going through, there was a slide I seem to have recognized, but yeah, it was not in your presentation. It was probably afterwards, one of the slides later. Anyway, no. The question is.
Strains. Yeah.
Apparently, have grid cells as well.
Yeah.
So
this is what we currently work on. This is what Lydia is now implementing and now testing.
Yeah.
We try to get as much of the same kits that we had for this study.
Yes.
With MR, with grid cells. This is already, it again took us 2 years to get at a task, because the first attempts to get a virtual reality task for 4-year-olds failed because it was too scary. We
Yes,
started
exactly,
with a
that's-
space
yeah.
environment, and space was scary. Now we have a magic forest environment, which works much better, but it took us 2 years to develop it, so we are a bit behind schedule here. But indeed, the idea is to get at grid cell signals. Colleagues from Leipzig did this already, and they showed at least in older kids there is some still change in the grid signal. If you think about the parallelism between mice and humans, you would expect that this is earlier.
Exactly,
So we would like to get as early as we can, currently 4-year-olds. So we adapted this task, and this is our magic forest task, where the kids try to find the nuts for a bear and a badger, and we have a turtle which serves as a movable landmark. This is one of the examples where you need to think about how kids think.
Yeah.
We have here a landmark which stays stable within a block, but changes across blocks. So we needed a cover story to have something that is fairly stable but can change. So we came up with the turtle. The turtle is slow. It needs some time to move. So within a block, it's natural to assume that it's at this point, but it's completely imaginable for 4-year-olds that across blocks, the turtle has moved to somewhere else. And we increase this by showing how the turtle moves.
Yes, very good. Excellent, thank you.
Okay, so let's thank Markus again.
Before you all leave, this doesn't have to be recorded. And speaking of recording, I want to thank a few people. And first and foremost, I want to thank Iman and Fanny for helping us with the recordings and everything. Thank you so much. I want to thank the design and computation students at Albert Lang for letting us having the conference here. I think this is really an amazing location, and it fits perfectly to what we were doing. So this is very, very great. And I want to thank a few more people. I want to thank my super cool DFG research group. It is just amazing to work with you. So this is really great. I want to thank Matt especially for organizing and for everything that happened before we met here. So thank you very much, Matt, for organizing this so professionally. We had more support. Susanne is not here anymore, but most of you were in contact with her, with helping with, I don't know, the German text ID and whatever, and helping us with the coffee and the food. So we couldn't have done this without Susanne and Adrian, who also helped here on the side. But of course, first and foremost, thank you all for being here. Thanks to all the speakers for these really interesting talks. We have a lot to think about, and I think our project will profit a lot, and I hope we all stay in touch. There will be a conference next year, which will be in Israel. We hope it will be in Israel. And in 2 years, we will have another conference here in Berlin. So let's stay in touch. And thank you all for coming. Thank you for your questions and the discussion. And enjoy the weekend.